Sofia University in Bulgaria. It's hosted and we at meet virtually online, but it is good. Now it's Beijing time, noon time, 12 o'clock, and I know because we are from this.
Because there is a microphone. Yes.
And what's your time?
Here is 7 o'clock.
7 a.m.
Yes, a.m.
Oh, very early. Yeah.
Early. But thank you for your hospitality because you changed the bit program. It's okay.
Good morning. Yes. I'll say Mahdi. Your time.
It's 8:30.
When? Mahdi, you're wrong.
8:30. 33.
Good time to work. I will recall it.
The time is good, but the day is now the holiday for the weekend here in Iran. Friday is in Iran.
I hope you'll get through soon.
Maybe my kids are the meeting.
Good, thank you. News from Brazil.
Brazil, it is an hour in
8 p.m. It's in line.
no, Friday.
Friday. Okay, yes.
Yeah.
You are in the time. Yes, maybe 3 hours later you have to go to sleep. And
Yeah.
wait,
Yeah.
Baruch. Baruch. Yeah.
I cannot see him.
Okay, I will him later. Anyway, and online to me, it's our first time to have these all professionals from continents of the world, and right now it's good for us. Is even in China we don't have to meet together. Such as now I'm in the university. I'm 1.5 hours from the university in the company. Some technological improvements. My students, most of them are in the lab working in the university. And I invited Professor Sun from Beijing and Professor Ye from Chongqing, and different China is such channel.
I'm Sally from.
Okay, we now see each other. That's so smoothly for can we just stop all the videos and I give a few minutes to Professor. Now the bear from Bulgaria and see what she can deliver to us. Let's stop the video and makes it more smooth.
colleagues. I'm pleased to welcome you to the International Symposium of the Researches of Medicinal Plants, which is kindly hospitality of the Huazhong Agricultural University of China. I hope pronunciation will. Professor at cooperation between Agricultural University and so claimant for on a bilateral investigation of the resources and cultural traditional medicine between China and Bulgaria of the Belt and Road Initiative countries. It was as a response of the co-regular exchange program for China and Bulgaria Cooperation Committee of Science and Technology. It is jointly made by the Ministry of Science and Technology China and Ministry of Education and Science of Bulgaria, National Science Fund. I would like to take this opportunity to our two countries and of the organization that mentioned above and for their support. The keywords that have brought us together today are medicinal plants and recently World Health Organization estimated that 90% of people worldwide try to have a medicine and some aspects of their healthcare needs. According to the same organization, around 21,000 plant species have the potential for being used as medicinal plants. And over the past 2 decades, there has been a tremendous increase of the use of herbal medicine. However, there is still a significant lack of research data of this field. A glance throughout the presentation for today reveals amazing diversity of scientists from different parts of the world, different scientific points of view of medicinal plants. And symposium works as this provide a valuable opportunity for research scientists to share experience. And I'm sure you will have fruitful and rewarding exchange of knowledge and today. As we and look forward learning about the outcome. And now I see a video about donation of our friends from China. And I wish to say on behalf of me and my colleagues, and I would like to thank you and to all China friends to your donation of this complete set of Materia Medica and China Pharmacopoeia. I wish to say that it is really very important and timely gifted support for us, and we highly appreciate it. And colleagues, I wish to say a few words about where is in whose hands is going the available books. The University of Sofia is the oldest and in higher education institution of Bulgaria, and it is founded more than 130 years. It's the first library in Bulgaria. In contrast, our research group, you see colleagues here in this meeting, research group of pharmacognosy, which are member of Faculty of Chemistry and Pharmacy. We have only 10 years history, which is starting with the beginning of the resumption of education in pharmacy in Sofia University. And as a research group, we have many challenges ahead of traditional medicine. This is from our article till now in the field of ethnobotany and ethnopharmacology, as well as presentations you will hear today. For our students, we the first and only so far a lecture. to assure you that the knowledge which was in these books was accessible and used by scientists and students. And last but not least, I believe that this is the basis for our future cooperation. Thank you again for your donation. Thank you.
Thank you. Thank you for your comments and the introduction. This is very good. And right now, my student, he is using the video. We can see the books. So a two series. One is the white one, combining material medical. It is a very old connection. It's about 500 years ago, and practitioner in China, he wrote this one, and it's very important collection. And I will have a talk late few hours later. And for this one, and one is China Pharmacopoeia, and we have 6 volumes. And the first volume is Chinese medicine, and each of the volume, I will send 2 collections to Professor Annie, because we have this funding. I hope in the future I can donate to other professors as well. And all these are in English, so everybody can read and know how we do traditional medicine in China. This is a good chance, and maybe in a few months I will ship this one to you, to Bulgaria, and I hope you can enjoy it. And now the first session we hear, and let's go to the lecture. And the chair for the first is Professor José Carvalho, and he is a well-known professor in traditional medicine. I met him coming to China once, 2019, and now I'm helping to applying the China scholarship in China for experts, and I think we'll have a chance to invite him coming to China for a few years, and if we got this one, it will be very great. Now, Professor José, it's your turn.
Okay, thank you. At the moment.
José.
At the moment. Me?
Not well. That's the best.
You me? At the moment.
Okay. No. You may restart again.
Now.
Please speak to José.
Yes. Welcome everyone. Good afternoon. Here in Brazil, it is 1 now in. It is a great pleasure that you will be coordinating the first session of this meeting. It's a symposium that brings speakers from China, Bangladesh, Russia, Iran, Brazil, and Bulgaria. And I want to thank all the speakers, especially Dr. Shuedo, who organized. So to begin, I would like to call Dr. Wei San from the Institute of Chinese Material Media and Academy of Chinese Medical Science. Everyone will have 30 minutes to show. Dr. San.
Dr. Sun is around.
Okay. I will share my presentation. My presentation. Can you see my presentation PPT already?
Yes, well.
Okay. First, welcome all the guests from all over the world to attend this meeting. My name is Sun Wei. I come from Institute of Chinese Material Medical, China Academy of Traditional Medicine. My topic is herbal genomics, development, and application. I spent some time to do the current investigation of genomics. Already traditional Chinese medicines. First, I gave a list about a famous book named Ben Cao Gang, written by a famous pharmacologist in China. Includes about 10,890 kinds of plants and animals included in this book. And according to the latest investigation, about 1,200 species were included in the book. There's different kind of plants and animals. On the Chinese medicines, have some strategy to investigate the herbal mice. We provide a display herbal genomics, coding the life of herb, including model herbs, herbalism, breeding, herbal synthetic molecular identity, and herbalmic data bank together bridge the gap between the traditional medicines and international economic studies to review the genetics and biology of herb. In our life, decipher some genome, famous plant and genome, including and some, for example, the Paneth ginseng, Celeron, Horiza, and Ground Pegulata, and Segelia Sinica. And a lot of medicine has been deciphered using genomic tools. I will introduce some papers published recently by our lab. This paper about the famous medicinal mushroom, fungi, Ganoderma lucina. We decided this plant, this fungi genome, this genome is the first medicinal model fungus. Some famous newspapers report our finding, and in our lab we use genomic tools to decipher the genome and to see the trisapinoid metabolic pathways. A second example is another plant. It's an Asian Chinese plant named Cediado Tamaris indica. This plant has some unique properties because this plant can resist the drought. So we use the genomics method to decipher the genome. The genome size is 301 MB. And these plants have the high hazardity above 2%. After the sample genome, we annotate these plants on the genes. We found 20,760 genes and contain 20% transposons. Next, we use the law gene loss and gene expansion analysis. Compare this plant, the crop plus genome with a published crop plus gene. We found 7 gene chloroplast completely absent this plant. So we published this and tell us about how these traditions can be resistant to drought, and we strategy to find a best way to promote some famous medicines such as licorice, salva, to resist the drought. The third story about famous famous plant. This used as is Sechamia nacania. It's a native species. We've reported this plant genome using the nanopore technology and to use a genomic method to introduce to interpret the diversification of certain biosynthetic associated genes. And we also study a big basis named endographis data. We can complete this. The genome size is 2 CT9 and B, and the least genome include 25,400 Chinese protein coding genome. And then we also estimate these plants with their ancestor version time. In order to decipher the important content named the endographite is a type of compound. We analyze the gene and systematically to investigate some important items families. We use the metabolome method and some item catalysis method to some key genes involved in the endographite pathways. The arrow indicate the genes. Last, we highlight another famous traditional medicine called ginseng, Panax ginseng. Its roots contain a lot of tricyclic compound named genocide. We use this genome project has a competition with others. We first report this genome. Genome is a huge, large, and complex genome, and this presentation of the genome annotation and the result. This ginseng plant has the highest proportion of the repeat sequence in the reported angiosperm, and numerous genes related to tricyclinoid metabolism has been annotated, including P450 gene transporter and the transcription factors. This is the detail of these papers. We use the decimas to review the distribution of genocide in the foods, and we annotate 31 genes involved in the genocide, and we use a lot of experiment to decipher to study these tricyclinoid pathways. presentation showed a special fungus type using the assisted hybrid breeding method. Above presentation is some genome and block metabolomics pathway in study. Next, I will introduce, based on this genome sequence, we established a system or assisted breeding system with the molecular markers, high quality Chinese medicinal materials. We screen. First we build high quality, high yield population and screening SNP by genome sequencing. Next, we confirm the DNA mark and combine with the phenotype. And last, we use this DNA marker to help us to identify the high quality or low quality population. We also finish the new breeding to sub-base some land race and land race to Beijing And we also create some positive markers to identify other panic species. This is image plant named Artemisia luna. We use the markers and metabolomics content tag together to screen land race. And after 3 years of propagation, we have this plant. Okay, this is live information on genomics and assisted genomics tools to breed. Next, I will introduce decoding. We use the genome sequencing information to select some base markers to help us to identify the Chinese because in the market a serious confusion of Chinese herbal medicines, very difficult to distinguish the true form, the form of Chinese medicinal powder, and some slide. So in our life, we first postulate the two ideal markers to identify the medicine plant species. We confirmed that these two had the high efficiency and the stability in the identification of the traditional Chinese medicines. After we collecting more than 2,172 samples from and 4,385 samples from plants, flower, fruit, seed. After lot of plant identity, we build this system use ITs to help us identify genes. First, we collect 11 solvents species. They belong to 270 families, and we invite some experts from to help us identify collecting plants from Kew Gardens, from the China Academy of Science, and all the sample numbers is more than 10. We invite some governmental experts to help us check these systems. And last, we build a database and some informatics tools to help us to identify these genes. These are some examples. We are also publish some book and build SOP method, and also we provide some identification for some company. Standard in barcode Chinese medicine was incorporated in the Chinese pharmacopoeia. Also, we train some experts from pharmacopoeia and train some staff from the Hong Kong Health Department to finish this system to identify the herb license. These examples. This is another examples. We are barcoding methods to identify some gene species. Okay. Next, we will continue to update the herbal genomics project, and we try to bridge herbal genomics with other omics. Finally, publish local breeding system for herb. Okay, thank you, all the listeners. Okay.
Thank you, Dr. for beautiful presentation. I would like to say that my students are streaming on YouTube, and now about 25 is watch presentation. Thank you. And thank you. Now I would like to call for next presentation Dr. Kumar from University of Chittagong from Bangladesh. Dr. Omar will speak about from the field to the level new learning of Bangladesh traditional medicine. Please, doctor.
Thank you, Jos. Can you hear me, please?
Thank you.
Okay.
Symposium organized by Dr. Ho Shuibo, Professor of Huazhong Agricultural University, China, and other guests, especially from some professor from Bulgaria and his Chinese professor, other students. Please take my warm greeting. And definitely this one is great symposium about the traditional medicines. So today I want to talk about the Bangladeshi traditional medicines. Let me share my screen first. I think my slide. Okay, can you see?
Yes, very nice.
Okay, thank you. So the topic of my talk is from the field to the lay and new era of Bangladeshi medicines, which we are calling EM. So the
Beautiful this slide.
Sorry, Professor Jones, can you repeat again?
Beautiful this slide, please.
Okay, run, right. Now, okay. Thank you. Sorry, I didn't get it. So this is the topic, and there are background information I have put it here. Actually, there are many types of traditional medicines in the world. So these are, all of you about these, the categories of traditional medicines, like the world renowned. There are some traditional Chinese medicine, Ayurveda, Arabic, Unani, etc. So our status, the status of traditional medicines in Bangladesh, like we have 15 government educational institutions, 10 Unani system educational institutions, 5 in teaching private system also. And we have more than 400 big and small manufacturers of herbal medicines or traditional medicines in Bangladesh. So some products are here, like the Carmina, the herbs, like this one is so good, this one also from one plant, etc. So traditional and complementary medicines, there are so types of medicines are there. Basically two types. One is medications and another one is non-medications. So medications, we focus on medicinal plants in our country. In our research group, we are doing research on only medicinal plants. So these are the status of medicinal plants in Bangladesh. Previously, there are many types of researchers. They are reported that medicinal important plants about total 1400. Some of them are telling that this is 55, some of them reported 450 species, or some of them are told that 330 species. So I have started my research on this field from 2006, and let's see what happened and what will happen after that. So this is our outline of the study. We are doing the first thing, painting the informations of the field, and we analyze using some qualitative thesis. We are going to study, there are many branches. One is anti-malarial activities, another one is pharmacos study, then another one is tumor and cancer activities, and phytochemical studies. So these are the objectives for the study. We just telling you, we want to know the each and every medicinal plant species of Bangladesh, and we want to find those medicinal plants, and we want to justify traditional treatment system through in vivo and in vitro studies. We are doing that. On the other hand, beside biological studies, we want to know actually what kind of active compound we need to and let's see. So the one is this is the first one, ethnobotany, and we know all of us that what is the meaning of ethnobotany, how can we define this? In Bangladesh, basically we focus on indigenous communities because they are practicing these types of medicines properly, and we have 35 indigenous communities of Bangladesh in total, but we have 2 indigenous communities in Chittagong Hillcrest. So this Chittagong Hillcrest, where I am living, and this really zones, how a high mount there. So there are many indigenous communities who are living nearer to the, and they're taking medicines, they are preparing by using the medicinal plants. They are not like modern, still they are not receiving modern treatment or modern medicines. So that's why we targeted them to collect or document the medicinal information from them. There are some methods what kind of methods we actually using to document this. There are some field, this is called field interview. I think all of you know about it. And this is group discussions, another group discussions, field interview, plant interview. There's some plant interview. This one also group interview, plant interview, and plant interview. This slide I can say about. You can see here, this is the 2006 where I am taking informations from one traditional healer named Ram Das, and his age was 101. I mean more than 1. So I have collected, as I remember, I have collected 36 plant raising informations from him at that time in 2006, and after 2 years he died. So important things that I was there in review. I have taken it and I have collected 35 plant using information from. After 2 years, I mean 2008, I went to there again, but I didn't find that person because he died. So these 35 informations now we are trying to find the treatment, traditional treatment system or analytic research. And we are also trying to document all those medicinal plant information before losing or before lost like him. This is the very important things that we lost like this information we not collect from today. So so this is the studies, and then summary is here. Our study about that. What is the status of medicine now? 2006, we started this work, and we have documented 1,479 species as a medicinal plants from Bangladesh, and this is the first time we have done this. And more than 2,000 plant information we have. And still this research is going on. So what we have done? So what we have we prepared 2 data and published these databases. One is plants database of Bangladesh, and here we just want to include or enlisted 1,479 recorded medicinal plant informations along with their other botanical, taxonomical, ecological, and phytochemical, all kinds of information. Already we have enlisted here more than 900 species. This is another one. Laboratory online lines that we have used. And we try to know actually this some anti-cancer property. So these are the way of biosecurity fractionations that we have done. And we have here some compounds, and we have done some biological assays and the morphological assays on me to I just put it here to show that actually how we are doing cancer activities. As here we wanted to know about this factors. Is there any by annexing the test? There are some Western blot. We try to know about some antibody, and we have in the PCR2, PCR2. Here some respiratory. This one is negative respiratory. To be summarized those results here by comparing some but this is the mechanisms compounds, and so these are the about activities. Still we are doing some plants against secondary cell lines, but these are not public. Some after 2 or 3 months we have published it. Another one is pharmacological. So we have published here many articles. This one is about the one species Huazhong caustica. This plant for the eye irritations, inflammation, diseases, scars, and all. So we tried to justify this condition, and we have recorded these conditions earlier about on this species. Our results showed strong and significant and those compound through the CMS technology, and we saw all the documented five compounds with 49 of this. This one has been published in Journal of Pharmacology. This is another one about the one. If you also use for the treatment from diarrhea, anxiety, the diarrhea, and things and significantly support the traditional steps. We have published in another journal, Molecules. This is another one. This is another plant. This is Tetrastigma phylum. We have tried to know about actually used for the diarrhea. Actually, this plant showed good results and significantly reduced diarrhea restored rates. And this one also we have published in Molecules in 2020. So this is another one. My previous year, just previous speakers, it showed that the environment. This one is very important to identify the plants, medicinal plants. First time we have done 20 medicinal plants of Bangladesh medicinal plants. We have finished this, but this we are writing manuscript right now. We have finished 20 medicinal plants to know the molecular interventions. We target to purport, of all record species that is 479 Bangladesh medicinal plants. We want to do this field barcoding to know the actual proper. Then the pharmacological activity. This one is also very important things as the neuroscience technology, one of the big pillar research areas, neuropharmacology. So we have some plants, recorders, some plants that they are using for the treatment and do research. Is showing topics we have explored medicinal plants. We have released 5 plants experiment. These are the some results. This is one of is unpublished. Doing like also this neurofarmer project. Then the program with the collaboration of under 2010 we have one plantation garden where all species to preserve and we want to plant all these plants, 179 plants for researchers. Anyone can go there. This is very and anyone can go there and can collect the plant parts for their research. Patients. We have done some genome sequence for COVID-19 SARS-CoV, and by this time we have published this one also. Another one is also still we are preparing for publish. And here this is the time in our city. I mean, we have done genome sequencing of 33 samples. We have the COVID patient. Basically, this we have done this one to know actually what kind of mutations, what kind of virus nature, from where they enter to our city, etc., etc. Besides, we are also trying to do some experiment on SARS-CoV-2 to know is there any plant that gets active against virus or any plant expert. So we are doing some initial screening now, and hopefully we will have some good results after a few months. The project is ongoing called mosquito repellent plant. You know, in our country, especially in Bangladesh and other regions, China, I have seen there are so many mosquitoes. So the mosquito people are using some chemicals. Those chemicals are harmful for the nature, not only for the human being and other insect, other creatures. So they are using to control the mosquito larva higher concentration of harmful chemicals. So we are doing some research right now. Now ongoing research that so many plants we have recorded from the indigenous communities. They are using this plant to control the mosquito. So we are trying to know about now what kind of plants are active or what kind of the larva of the mosquito. So the project is ongoing. Hopefully we'll get some results after 3 months. So the novelty award study is we have done many first modern legal study in my country. Yes, some experiment in the legal article we have many. The conclusions is like that. This is still a process where is look towards the hands uses it was lost forever. Our we hope plant diseases and our our research group recorded 5,000 Islamic plant species with authentic identifications in Bangladesh. Currently, we are trying to plug the and total flora. I mean, we included there. Welcome to my country and my University of Chicago. And thank you very much. If you have any questions you can ask me. Thank you very much. Professor Jules.
Okay. Thank you, Dr. Omar, for beautiful presentation and your work is beautiful. It's the best for develop new products for your country, Bangladesh. Now I would like to call Arlindo Cesar Pereira. Arlindo is PhD candidate from University of São Paulo, Brazil.
Excuse me. Can I have a question? Hi, Farouk. Nice to see you here.
Yeah, I'm here.
So, just one question. Can you really the medicine that can be commercialized or in China? This is very important. Sure, there's too many things we can do, but we do important things. Focusing on things can be commercialized. What people are treating kind of diseases. Can you hear me?
You are asking about mosquito plant?
No, not that one. I mean, any potential to treat disease in commercialization, and in this, I have to think about the yield, quantity, and the way how much you can you get from the plant, and this is very important.
Yeah, definitely. Some of the let me see the result.
I have to say the pound from plant to the microbes. This way will be very hard.
Yes, that was the difficult.
Yeah. Maybe can some other inflammation, but not in microbes. Okay.
Okay.
I guess you can search, and in the last years there's no any kind of plants, all from microbes. Okay.
Okay.
Yeah, that's juice.
Thank you, Dr. Omar. Now would you like to call Arlindo Pereira, that is the candidate from University of São Paulo. Pereira speak about medicinal plants with anti-epilepsy potencial.
Yes. Well, let me share my.
Okay. Yes.
Okay. I want to thank the organization of this event for invitation. My name is Arlindo Cesar Pereira with anti-epileptic potential. Well, I'm from the University of São Paulo, where I do my PhD. But during my master's, I worked with epilepsy. To talk about start. This is one of the most common neurological diseases in the world. This happens because this causes genetic mutations, can be caused by traumas, can be caused by metabolic disorders and autoimmune diseases. And hence, it's a very common complication. Okay. 1 million people in the world suffer from epilepsy, which is around 1% of the global population. The disease is characterized by the occurrence of seizures, however, despite several about 1/3 of the patients do not respond adequately to the treatment at the first moment, which is 30% that do not respond to the treatment. And even among the patients that respond to the treatment, some of them come which is a very huge drawback of our current compulsive drugs. This situation called epilepsy public health. Also, there are some advantages with our anticonvulsant agents, such as cognitive. Just to cite a few of them. Actively, this shows the necessity of treatment approach. And we know that plants are long as source of bioactive about plants that help to the treatment of epilepsy. But why plants? Well, it's believed that compound extracted from plants morphine. After morphine, other schemes such as choline, digitoxin, and many others, as you may know. However, only between 5 and 15% of the higher plants have been screened for medicinal purposes, which means that a very great percentage of them were not assessed. Also, despite between 10,000 and 50,000 plants are documented around in the medicine, okay? means that there are several overlooked species in our medicine. In the Amazonian rainforest, is believed largely overlooked, with around 1,000 vascular plant species. Okay, in such a huge biodiversity, there's a big potential to bring new compounds with the potential to be used in treatment of epilepsy and several other CNS disorders. Okay. Now I will talk about some of the species that are used by traditional people in the Amazon rainforest that have the potential to be used against epilepsy. You know that there is a limitation of time, but the first one is Petiveria aliacea, okay? So the popular name of this species is Mucuraca, Guiné, Ipi, and Mansa Senhor, which means to tame the master. His name, to tame the master, is because the old days, these leaves used preparations of this plant species to calm down their masters, and then this name came, to tame the master, Amansa Senhor. Some of the ethnopharmacological uses of this species are to treat anxiety, to treat some kind of pain, memory deficits, and some kind of seizure, okay? Well, here is the geographical location of the species. Today, the species is very widely distributed around the world, but some researchers believe that the origin of this species is here in the late America, okay? But today, this species is found in Africa and in India as well, okay? There is one study that was published by Lima and collaborators who tested antinociceptive effect of this species and the anticonvulsant effect. Talk only about the anticonvulsant effects to the theme of our presentation. And the authors tested the infusion from the roots. Okay, they tested in mice 3 doses, 500, 1,000, and 2,000 milligrams per kilogram. They tested these doses against seizures induced either by electroshock or by intranasal, which are 2 very common methods to induce the seizure. According to the authors, the treatment would decrease the locomotor activity of the animals in the field tests, which is a very common behavioral test. This is a sign of CNS depressant activity, okay? This means that the plant preparation has a depressant effect in the central nervous system of the animals. As for the seizures, the authors reported that the treatments increased the latency to seizure, increased the seizure duration, okay, which are 2 positive outcomes of the treatment. And very importantly, the seizure-induced mortality decreased with the treatment as well. And the dose with the best results was 2,000 milligrams per kilogram. Some years later, another group of researchers performed another They tested different preparations of vetiver in several behavior models using mice. Was performed by Gomes and collaborators. Okay, the authors, as I mentioned, several using acetate, the hydrochloric extracted from per kilogram in mice. They test the authors reported that the treatments decreased the motor activity in the animals and increased the pentobarbital-induced sleep time. These two phenotypes are signs the present effect, which is in accordance with the previous report that I mentioned, this by Lima and collaborators. In addiction, no significant effect on motor coordination was detected in the Rotarod test. As for the seizure, it was observed in the pentylenediazole-induced convulsion that the treatment also increased the latency to the convulsion and decreased the time of convulsion, which is also in accordance with the collaborators. Okay, this plant species is very promising for a future product to be used against epilepsy, called Vetiver liacea. Now to talk a little about eclectic. It is found in the Amazonian forest. However, it's not think of the Amazon rainforest. As seen in this map, this plant is found in the Amazon, but is found in the northeast of Brazil. This plant is from the family Asteraceae and is popularly called Mata da Terra, which means first mata. And some of the ethnopharmacology of this species is to treat some forms of pain, due to its sedative effects. Among others, formed by Souza and collaborators, these authors tested 3 different activities in the essential oil of this species. The antinociceptive activity and talk again about the anticonvulsant activity due to the theme for presentation. From 15 to 400 milligrams per kilogram, and they tested in the anticonvulsant assessment, tested the treatment pentylenediazole-induced seizure or seizures by strychnine. According to the authors, it was observed statistically outcome in the anticonvulsant activity assessment, and the mortality caused by the seizures also decreased in a dose-dependent manner. Those tested above the mortality is a very positive outcome. Again, with this species, this time is Egletis posa, okay? The essential oil. Now, we'll talk about you. And there is one study that was published by Carlini and collaborators. They tested two preparations, the essential oil and the hydrolate from the seeds of Licaria puschuri, okay? They tested for several psychopharmacological effects. I will mention the effects on seizures. The authors tested the essential oil and the hydrolates from Licaria puschuri in several psychopharmacological effects in mice with doses ranging from 50 to 800 milligrams per kilogram. Well, the essential oil and the hydrolates could decrease the animal's motor activity, which is evidence of CNS depressant activity. However, only the essential oil could decrease the electroshock-induced seizures. The hydrolate couldn't decrease the seizures, which means that only the essential oil had anticonvulsant effects. Notably, this study assessed the chemical composition of the preparation, which is very important because we need to know which molecules are involved in such activity, okay? According to the authors, the oil was composed mainly of saffron and cineol that together represented more than 75% of the oil composition. Well, there are cineol, 1.8 cineol is a monoterpene. We know others monoterpenes that has anticonvulsant activity. In our research, we worked with a monoterpene, as we'll talk about later, that also had anticonvulsant effects. And the last species that I will talk about is also an endemic species that is found only in the Amazonian rainforest, and it's called Passiflora lata. This species from the family Passifloraceae, and it's from the same genus of Passiflora incarnata, which is the popular passion fruit. Passion fruit here it's called maracuja, the popular name, and this species is also called maracuja because they are very similar. But only this species is endemic from the Amazon. There are also some ethnopharmacological uses of this species, such as sedative activity and hypnotic activity. There was one research that was carried out by Poga and collaborators. These authors tested the fluid extract from the leaves of Passiflora lata at two different doses, 75 and 150 milligrams per kilogram in mice. The treatment significantly increased the barbiturate-induced sleep time, which is, as we mentioned before, a sign of depressant activity in the central nervous system. Both doses showed anticonvulsant effect by increasing again the latency to seizure and decreasing the mortality against the pentylentetrazole-induced seizure. And this was observed very interestingly in a dose-dependent fashion. Well, also the animal's spontaneous motor activity was decreased due to the treatment, which also means that the product has CNS depressant effects. Okay. Well, as you can see, there are several species that has the potential to develop a future new anticonvulsant agents, or a potentially lead compound that could be used in our arsenal against epilepsy. However, there are some limitations with the current studies. For example, many of these studies, as you may have noticed, are very old. They have old methodologies and could have more important parameters assessed. As I mentioned, for example, the chemical composition. Only one study performed the assessment of the chemical composition, which is crucial for to assess the activity of a medicinal plant. Okay, and as we mentioned before, there are several plant species that has the potential that are used in ethnopharmacological study, and only a few of them were tested experimentally, which is a huge potential for new research. It's a truly golden mine for the research in the treatment of epilepsy and also other CNS disorders. Okay. In our laboratory of Professor José Carlos Tavares, that was my teacher, my professor during my master's degree, I worked with epilepsy, and we tested some products against epilepsy, mainly those that are derived from plants. For example, I tested the effects of peryl alcohol against epilepsy. Peryl alcohol is a monoterpene, and it's found in several species from the Amazonia, but not from the Amazonia. Also, several other species from the citrus genus of the citrus fruit of the family Rutaceae. Okay. In this research, we tested the effect of peryl alcohol against pentylentetrazole-induced seizures using adult zebrafish, which is a very popular model in neuroscience today. Well, we observed that here is the structure of peryl alcohol. Peryl alcohol is also used in the treatment of glioma, which is a type of brain cancer. Okay, in our study, we tested in zebrafish, in adult zebrafish, and we observed that the treatment, we used 3 different doses, which were 10, 50, and 100 milligrams per kilogram. Here we observed that the frequency of seizures decreased in a dose-dependent fashion. Okay, this first group was not treated, and these 3 different bars represent the 3 different doses of peryl alcohol, and we can see that the frequency of seizures per group decreased in a dose-dependent manner. And the same occurred with the loss of posture, which is a sign of seizure severity, and also the immobility, which is also a sign of seizure severity. They also decrease in a dose-dependent fashion. Then we assessed the animals individually for seizure duration, the number of corpuscles swimming, loss of posture, and immobility. In the seizure duration given here in seconds, it was observed, here we see the negative control group that was not treated, and the 3 different doses of peryl alcohol. Here we have the vehicle control group that did not receive pentylentetrazole, and here we see the control group that was treated with diazepam, which was the control anticonvulsant agent. In the number of corkscrew swimming, which are when the fish swim in a circulate way, it is a typical feature in the zebrafish convulsion. We observed also that there was a decrease in a dose-dependent fashion. And the same with loss of posture and immobility time, that are two signs of seizure severity. Hence, in our laboratory, we search for new compounds that could be whole plant products such as extracts and essential oils, or isolated compounds such as beryl alcohol against a range of disease, including epilepsy, which is a very important topic to be researched, to be studied due to the cases of refractory epilepsy, okay, as I mentioned before. Well, these are my references, and I want to thank you again for the organization of the event, and thank you very much for your time, okay? Here is my name and my email, and nice event for everyone, okay? Thank you.
Thank you. Thank you. Thank you, Arlindo. Thank you very much for excellent presentation about this theme, very, very important for you. Any questions? Then
I have one question.
Okay, please.
Okay, thank you very much for your nice presentation, Arlindo. Why did you do the adult zebrafish? Why don't you use larvae for the anti-epileptic activity? You're mute. Okay, now responding, that's a very relevant question. Why use a zebrafish? Well, it's an inedited study, okay? It was this activity of beryl alcohol was never assessed in the literature before. Hence, we were not sure that the treatment would work, okay? Then we decided to use first an animal, organ animal that had a better cost benefit ratio. You understand? Zebrafish, for example, you can use 10 or 15 or 20 animals per group, and you can make 10 groups, okay, with 10 animals in each. And this would be very cumbersome to do with rodents because they are very expensive. And then we can use zebrafish to do a preliminary study, and then we can use the rodents to validate our results. You understand what I mean?
I know, but you use the adult zebrafish. It's normal to use the larvae.
Okay. There is a reason for this too. Okay, it's because the larva of zebrafish do not have the complete central nervous system, okay, and also they don't have the blood-brain barrier, which is very important in the pharmacokinetics of anticonvulsant agents, because they need to cross the blood-brain barrier, and the larva zebrafish doesn't have the complete blood-brain barrier. Then these are the two main reasons to choose the adult zebrafish instead of the larva, okay? Because we tested through gavage, and also it is very difficult to do gavage in larvae zebrafish.
Well, you did the gavage to adult zebrafish.
Yes.
Interesting. Good luck.
How big is the larva of the zebrafish? How big? Hi. Arlindo. So
Here.
how big is larva?
Thank you very much, Arlindo.
What's the price of the
Thank you,
larva fish?
Sorry, I can't understand.
How
Please
big?
repeat.
What's the size of the larva?
Size. Okay. We use it. Adult zebrafish. They have around 4 centimeters in length. Okay, 4 centimeters in length. At the adult zebrafish.
Oh. Okay. Good.
Thank you, Arlindo.
Thank you very much.
Now I would like to call Dr. Xiaoli Ye, professor from Softs University. Dr. Ye speak about the development and the utilization of Coptis chinensis. Please,
Okay.
Dr.
Okay. So let's open my PowerPoint and then share. Can you see the picture clearly?
Yes.
Okay. Okay.
Yes.
Good morning, everybody. I'm Xiaoli from Southwest University. So today, I will talk about the development of the utilization of Coptis cerebrus. This one, I have been working this traditional Chinese medicine for about 20 years. So today I will talk about this. This is the main contents I will talk about. So the first one is the distribution of the species of this one. So this is the distribution of the Coptis cerebrus in the world. There are 50 species in the world. Just this one. They are mainly distributed in North America, Japan, China, mainland, and Taiwan. There are 60 species and one variant in China. And these three species are famous medicinal species. So they are recorded in Chinese pharmacopoeia. So next, I will introduce the differences of these three species because those of them are commonly used as a traditional medicine. So this one is a Coptis chilensis. Sometimes we call the William, and this one is a Coptis, the Deltodia. We see Yalian, and this is the Coptis Deltodia. We see Yunian. This is the plants. the whole plants, there are some difference. So this is the distribution. From this form, we can see that distribute in different places, such as this one. Many distribute in Hubei, Sichuan, and Shanxi, and both all of them are grown on the mountains, such as this one. It must grow on the mountains at the altitude about 1,000 and 2,000 meters. And this one many distribute in Sichuan, such as Omei and Hongya. The altitude is higher than this one. It must be grown on the mountains at the altitude 1,500 meters. They're often cultivated, and this is the Yunnan. Many distributed in northwest Yunnan and Xizang. They must distributed in very high altitude, about 2,400 meters above the nettles. This is a reason. The Coptis species many use the rhizomes. These three have the significant differences. Is genesis. We can see the rhizome has many branches, curved, and always gathered into a cluster, and shaped or formed like chicken feet. So the yield, because all of these plants will be grow 7 to 8 years and then harvested. So we see the yield always means 7-year yield. So the yield per moon is about 300 kilograms. And this one is the Reunions rhizome. So this is the single branch, and like the sickle worm. So the yield is lower. It is only about 70 kilograms per moon. And this one also has single branches. Then the final, the shape is like scorpion tails. So the yield is the lowest, is about 20 kilograms. So this one is the main species for medicinal uses. Next, I will introduce the Coptis, the major species, Coptis chilices. This is the red one. Is a major agricultural region for the Coptis chilices, and this is Hubei Lichuan, and this is Shizhu. So, because the Shizhu is mainly one of the major planting regions for this species, and because there has the GAP base, that is good agriculture practice basis. It is established by government, and also here is genetic resource reservation centers for keeps the Coptis species from over the world. You can see here as reserved many species, and this one is a planting base of one farmer. It's because planted on the mountains, so you can see a lot of mountain, the Coptis and the chilices. So this mountain belongs to just these farmers. It has about 500 rooms, so he is a planting expert. We can say he is. So next, I will introduce the utilization of the properties. Before starting, I will talk about some problems about the utilization of rhizome properties. First one is the cultivation. As introduced just now, as the properties is perennial herbs. Always need 7 to 8 years before harvested. During this period, the weeding is a time and labor-consuming work. Besides that, the disease such as root rot would seriously affect its growth, causing the yield decrease greatly. After harvested, the soil is not suitable for planting Coptis chinensis anymore. So if you want to use this one, you must plant other medicines or vegetables. If he want to continue to plant the Coptis chinensis, he must reclaim new lands to cultivate it. So this phenomenon is also continuous cultivating obstacles. So these problems make more and more farmers leaving home to find ways to make a living. Subsequently, the planting area will be decreased greatly. Therefore, I think it is very important to solve these problems to maintain a stable planting areas. This is and another question is the drying process, because the rhizome must be dry for storage after harvested. The most common used method is using the smoke of varying wood. However, the biggest problem with this method is that it is difficult to control the baking temperature and time. So the content of the active ingredients in this rhizome is lost about 20%. So I think this is also the big problem must be solved to find a way to maintain the quality of the rhizome during the drying coursing. This is true. And the other questions, such as the TCM preparations, I mean the preparation is containing rhizome qualities. Some qualities, quality standards of preparation need to be enhanced because some of quality standard is very low. So we just detecting one ingredient. For example, Jordan peel is used just use berberine. Is one of the active ingredient in rhizome properties. Use this one as the quality control standard. If the berberine content meets the requirement, it means this preparation, this peel is qualified. So some company will add some extracted berberine from other chipped herbs or synthesized berberine into this peel to replace this the expensive rhizome properties, because the price of the rhizome properties is about 150 RMB per kilograms. So because of this, so the TCM preparation will cause the efficacy of this weakened or lost. In addition to some of the TCMs, the effects is limited. So we want to enhance their utilization based on future resources. And the first one is called efficacy. You see, this book, is two famous book. This one is a compendium of Natura Medica. In it recorded 27 functions of rhizocoptes, and this is the Chinese pharmacopoeia. Also recorded 51 prescriptions containing coptes genesis, which can treat 73 symptoms. So, if we want to, there are so many applicants. So we need explicit which one is there. It's core efficacy and explore the mechanism for the development of new drugs. The last one is the utilization of the byproducts. This is the properties. So we can see here byproducts such as the flower and the fibrous root and the others and the wishes. So all of them are thrown away as waste. They will pollute the environment. If we can make full of them, the waste would become the valuable products, and the outcome of the farmer would be increased. So our team focus on these orientations, such as preventing diseases and continuous planting obstacles, and find a new way to get the high quality of the rhizocoptes, and also try to improve the quality of standards of the TCM containing rhizocoptes, and from which we can find some new drugs to do. Also, we can make full use of the byproducts. So this is the first example. We use the byproducts of the coptes chilices. This one is the flower. So we want to develop it. Before we develop it, we will analyze the medical ingredients and the nutrient components. From this map, we can see the inflorescence contained just two echinoids, pomatin and berberine. The total content is about 1%. It is far lower than the rhizocoptes. And also, except that it has some nutrients, such as the total protein is up to 26%. Also, there has the total amino acids is up to 16%. So, more important, we from the total amino acids find it is contained 7 essential amino acids, and the ratio of the essential EAA to total amino acids is about 39. It is very close to the ideal proteins. It is about 40%. So we think maybe it can be used to develop new vegetables or food. So next, we do some experiment to convert it. This is three to convert its the safety. We do the acute toxicity and the chronic toxicity. We think it is relatively safe. Also, we do some pharmacological experiments to confirm whether it has some has some pharmacological functions. We use diabetes rat induced by STZ and the hyperlipidemia rat induced by high-fat diet. Also, we use the fever mice by injecting gas. We use these three models to confirm the influence extract can lower the blood sugar and lower PC, TG, and LDC. Also, it can release heat. So we think it has potential to develop vegetables. And then we want to develop it as the tea, the flower tea. But because it is just one part of the traditional medicine, so it is very hard to develop it as a food. So there is a long way to do. This is the first one we want to do. Then another byproduct is fibrous coptis tilices, fibrous root. We also analyzed the content of the alkaloids. We found they had the similar ingredient, the components of the alkaloids. Just some of the alkaloids had the different content. So we also did some animal experiment to confirm it has the similar functions to the rhizome. So we developed it as a veterinary medicine. And last year we had get the two certificate authorized by Ministry of Agriculture and Rural Affairs of People's Republic of China. So if we have success, so we can get it, increase the value of the value. So also we use some byproducts as a materials and extract the alkaloids from it and develop it as the health products such as soap and toothpaste and some antibacterial liquid. Also have the cream to prevent acne. So I think this work is very interesting to do. The third content is the co-effect of the efficacy of the rhizome coptis. Maybe everyone know the rhizome coptis has various functions. Are to just focus on these four antibacterials, diabetes and complications, hyperlipidemia, obesity, and cancers in digestion systems. So next, I will introduce three of them as the example. This is the alkaloids in the rhizome coptis. Many reports have showed that these alkaloids are the active ingredients of the coptis tilices, except that especially including berberine coptis and palmitin, berberine, etc. Also, there have some other components such as lignin and oxide substance. So now we have established some technology to extract and purify these alkaloids with very high purity on a large scale. So we can use it for the experiment. This is the safety evolution for the different kind of the alkaloids from competent citizens. This is the lethal dosage, half lethal dosage, and it is very, very safe, relatively safe. And this is chronic toxic, 90-day fate toxic. Both the experiments indicate that they are safe. So we do some the pharmaceutical experiments to compare this one is alkaloids can improve the renal injury of diabetic nephropathy. And first, we extract the different alkaloids from it with high purity, and then we use network pharmacology and use the cells, methodial, and also we use the diabetic nephropathy as the models to screen which alkaloids can have the functions to improve this one. So we found epinephrine showed the best results can improve the diabetic liver. Then we use these cells and mice to verify the activities with different dosage, and then we use the transcriptome and gut microbiome to find the potential mechanism of this. At last, we get the information. We get some conclusions. We found EPI and berberine can improve the DN. Also, the EPI has a protective effect on kidney. This is a histonic observation. Also, we have found the potential mechanism. It is by targeting AGT, a signal pathway, to regulate it. The second one is that the rhizocoptes improved obesity and fatty liver by high-fat diet. First, we also extract the alkaloids, and then we use the two models. These models is a leptin receptor deficiency mice. So as the opposite models, and this one we will feed high-fat diet for 8 weeks, and to get the fatty liver and the nutritional opposite models. Then treated with alkaloids. This one will treated 10 weeks, and this one will treated 12 weeks. So the result showed that the alkaloids can lower the body weight and the fat weight. Also, they can lower some the serum TC/TG levels, and this is historical observations, it has also can improve that. So we found just this one here. We do different alkaloids. So just this one, getaridin, can show the best results to improve the obesity and the fatty liver. So we just focus on these alkaloids and to find the potential possible mechanisms. So the last one, we focus on the cancers. Many papers report rhizocoptes can prevent many various of cancers, but we just focus on the digestive cancers such as gastric cancer and liver cancer and the colon rectal cancer. Here, I just use this one as the example. The gastric, based on the statistics from WHO in 2020, the gastric cancer has ranked the first among all malignant tumors in China. So we just focus this one. Coptes, we found coptes can inhibit the proliferation of the gastric cancer cells, reduce its apoptosis and cell cycles, arrest in gastric cancer cells, and then we use the not mice as the model. It injected the cancer cells in it, and then we get the model. So we found it also the alkaloids can inhibit the growth of the cancer cells in this body. This is the tumor, and this is this pathway. So this is the alkaloids inhibited gastric cancers result. So next, I will introduce our future work. So I think these 5 directions are important. So our team will concentrate this work. Okay. Thanks for your attendance. And this is RMT. Thanks for our team's hard work. Okay. Thank you.
Thank you. Thank you very much, Dr. Ye, for presentation and full work. Beautiful, beautiful. This is full work, beautiful. Any questions?
Okay, thank you.
You have time or any questions?
What?
Then I would like to call Dr. Ivan Vinarov. That who is associate professor from Sofia University, Hungary. Dr. Sivarov speak about quality control of herbal substances from medicinal plants. Please, Professor.
Hello to everybody.
Yes.
Thank you, Professor
Tavares. Okay, next 20 minutes I'll talk about quality control of herbal substances from medical plants. After a short introduction in the field of plant substances and ionic liquids, I'll present our results in the direction of solid liquid and liquid extraction of natural products from herbal substances and herbal drug preparations with quality control purposes. Let's start with the definition of herbal drugs. According to the European Pharmacopoeia and the European Medicinal Agency, herbal drugs and herbal substances are synonyms, and they are mainly whole, fragmented, or broken plants, part of plants, alga, fungi, or lichen, in unprocessed state, usually dried form, but sometimes fresh. Certain extracts that have not been subjected to a special treatment also are considered to be herbal drugs. In each monograph of herbal substance in European Pharmacopoeia, there is identifiable part, quality test, and assays for quantification of a biological active compound or group of compounds. UV spectroscopy, gas chromatography, and high-performance liquid chromatography, are the most used techniques for quantification of natural products and part of quality control of herbal drugs, respectively. But the problem is that in these assays, organic solvents are used for extraction of herbal drugs. That using organic solvents is risky because they are toxic, flammable, and require special equipment for working with them. The solution of this problem in quality control of herbal drugs is to replace organic solvent with ionic liquids. But here comes the question of what ionic liquids are. By definition, ionic liquids are salts which are in the liquid state at temperatures below 100 degrees. You know that sodium chloride is a typical salt composed of ions, but its melting point is around 800 degrees, and sodium chloride does not comply the definition of ionic liquid. The high melting point is due to high symmetry both of cation and anion. If we replace sodium cation with 1-ethyl-3-methyl-imidazolium cation, which has a significantly lower symmetry, the newly formed 1-ethyl-3-methyl-imidazolium chloride has a melting point of about 78 degrees, which is an order magnitude lower the definition of ionic liquid. The structure of ionic liquids can be very diverse. The cation is always organic. Some examples, and anion can be either organic or inorganic. Substituents of different length, structure, and functional groups can be introduced in both ions. In this way, according to some authors, more than 10 to 15 degrees of different ionic liquids can be obtained, which is a huge number, of course. Along with the huge number that can be synthesized, and the interest in them is great. If you do a search in Scopus with the phrase ionic liquids, you will find thousands of publications containing this phrase in its title or abstract. As it can be seen from a graph, in recent years, 7,000 articles per year are published each year, or more than 19 every day. And these are only the articles indexed in Scopus. This means that even you can't read all of them. But what makes this class of compounds so attractive? These are their unique properties, such as negligible vapor pressure, non-flammability, high thermal and mechanical stability, electrochemical stability, tunable properties, recyclability. Some of them have low toxicity, and their biocompatibility and biodegradability, which determines their use as alternative to conventional molecule organic solvents in areas such as synthesis, catalysis, electrochemistry, analytical chemistry, nanotechnology, and different processes and separation and purification. In 2006, the first publication appeared describing the use of ionic liquid as extractant for the extraction of natural product artemisinin. Dozens more examples of the successfully used of ionic liquids are as alternative to conventional organic solvents have been published. In 2014, Bogdanov summarized nearly 100 publications related to extraction of natural compounds in a chapter of book entitled Ionic Liquids as Alternative for Extraction of Natural Products. The main conclusion of this chapter is that extraction with ionic liquids provide significantly higher yields and reduction of extraction time and solvent consumption. When it comes to quantification for quality control, we have developed three procedures for the determination of biologically active compounds in herbal drug. We quantified exclusive compound galantamine in herb from Huazhong. The acetylcholinesterase inhibitor galantamine from herb of Lycopodium estivum and sesquiterpenes in roots of Valeriana officinalis. In all three cases, the organic solvents are successfully replaced by aqueous solution of ionic liquids in the extraction step. I want to give you an example of the advantages of ionic liquids over conventional solvents in extraction procedures. This is a standard sample preparation procedure for the quantification of galantamine in Lycopodium estivum, published in 2016. Briefly, weighted samples were macerated with ethanol for 72 hours and continuously extracted in ultrasonic pulp for 5 hours. Extracts were filtered and the solvent was evaporated in the vacuum, and the residues were dissolved in 2% sulfuric acid and defetted with diethyl 5 cycle of extraction. The aqueous layers were basified with 25% ammonia to pH between 9.5 and 10, and the alkaloids were extracted with chloroform 3 times. After evaporation of the organic solvent, the dried extracts were dissolved in 5 milliliters of methanol. Here used 3 organic solvents, and minimum 81 hours are required for sample preparation, right? On the next slide, you can see the proposed procedure by us. A known mass of plant material was extracted for 1 hour at room temperature with 5% water solution of butylmethylimidazolium chloride in open to the atmosphere round bottomed flask. The resulted extracts were filtered, diluted with deionized water, and was directly subjected to analysis. It can be seen that peak of galantamine here is separated from the other components, and its content in the herbal drug can be determined in less than 2 hours. This is 40 times faster, and no organic solvents are used. In the example still now, solid liquid extraction has been used to analyze natural compounds in herbal drugs. But when the quality of the herbal drug preparation should be determined, solid liquid extraction is not applicable because they are often liquids. In this case, liquid-liquid extraction should be performed. Instead of organic solvents, ionic liquid also can be used for liquid-liquid extraction. There are 2 types of liquid-based aqueous biphasic systems formed with hydrophobic ionic liquids. They directly form 2 phases, and with hydrophilic ionic liquids. The second type is obtained after adding cosmotropic salt to the aqueous solution of the ionic liquid by varying of type of salt and the pH of the medium. Controlled distribution of the target compounds in both phases can be achieved. In this book, we wrote a chapter where we describe in detail the advantage of their use in purification and isolation of natural phenolic compounds. In our research on the model system, we have shown for a series of natural coumarins that they can by controlling of pH, we can influence the distribution of analytes with respect to two phases. This can be used for their separation from other classes of compounds. We also performed studies with real extract from Lycopodium aestivum obtained with water solution of butylmethylimidazolium acesulfamate. Different cosmotropic salts in different concentrations are used, as the graph shows. Regardless of the salt concentration and pH of the aqueous phase, the alkaloids is preferably distributed in ionic liquid phase. This result was used to develop a procedure for isolation of galantamine in crystal form and regeneration of the ionic liquid. In another of our studies, we have shown that aqueous two-phase systems with ionic liquid can be used to concentrate samples. In the case shown, concentration of 160 times was achieved. In summary, I hope that with these few examples, I have been able to convince you that conventional organic solvents can be successfully replaced by ionic liquids for quality control purposes as extractants or of natural products from herbal drugs and herbal drug preparations. Thus, the procedures will be easier, faster, and safer. Thank you for your attention, and this is a drawing of our faculty. Thank you again.
Thank you very much, Dr. Ivan, for presentation and for his best theme that is very important for is quality control of herbal substance. It is very important any very content. But any question?
I have a question.
Okay, yeah, please.
Okay, thank you very much, Ivan. So I did not understand exactly your presentation. How do you get rid of the ionic liquid after the extraction? I mean, when we are going to propose it as a drug, or as a, so you have to prepare the kind of preparation for extraction of medicinal plant with ionic liquids. How do you get rid of the ionic liquid after the extraction?
Okay, just a second. I don't know what you see. Did you see this? Yeah. This is the procedure proposed by us, developed by us for isolation of guanidine as a hydrobromide from Guanidine flavum. Here we use a plant material and butylmethyl imidazolium acesulfamate for extraction of this herb. We perform 10-time cycle of extraction here, and after that, after separation of biomass from extract, here we can selectively extract guanidine from crude water extract by biphase system with cosmotropic salts, and after that we can regenerate ionic liquids here, and of course we can isolate guanidine as hydrobromide.
Which organic phase are you using?
Here we perform in this case. I think the extraction is performed with ethyl acetate, but also we can use, as is mentioned here, sodium carbonate. We can put in the extract sodium carbonate, and we can perform two phases. And the target compound will be distributed around 100% in phase rich of ionic liquid. And after that, this ionic liquid is not soluble in organic solvents such ethyl acetate, for example. And we can extract only guanidine. And after that, the organic phase is evaporated, and this is standard procedure. We dissolve guanidine in ethanol and put inside hydrobromic acid, and it crystallizes like salt.
Does this method accepted in the EP, I mean European pharmacopoeia, or this kind of standardized methods?
Which one? This for isolation of the guanidine?
Using the ionic liquids?
No, not yet, but I believe that this is the future, because can be found one or few ionic liquids, which can be appropriate for extraction of more than herbal drugs. And in this case, they are used, and after that, recycling will be easier.
Okay, thank you very much.
Thank you. Thank Dr. Ivan. Next presentation, I would like to invite Dr. Huazhong Hu from Huazhong Agricultural University. Dr. Hu is speaking about the compendium of Materia Médica, one short introduction in the medicine exchange between China and Iran along the Silk Road. Please, Doctor.
Hi, can you see me?
Yes.
Oh,
Well,
okay. So, today I have two talks. The first one is about the compendium of Materia Medica. And as you know, many scholars from China that will mention this one. And I found out that most of our international friends, they don't know what's this. So I will make a short introduction on this. And also, many of the Chinese medicine was from other countries. And I will talk about the very old traditional medicine from Persian and now Iran, along the Silk Road. You can, my name, last name is H.U. My first name, Hugo, but it's very hard to pronounce in English, so I wouldn't mind if you call me Hugo. Okay, I'm from Huazhong Agricultural University, and this Huazhong means central China. So I've shown this map to some of you, and in China now we have a national structure called the Belt and Road Initiative, and this one is made up of two strategies. One is the red one is along the old Silk Road, the red one, and it's through the land, and the other one is through the sea. And in both these two roads we have, our ancestors has crossed this over 10,000 miles in old times, and now we make different ways to boost the economic and culture exchange, and this is how we call this one. And in old times we go very far, and I will introduce from old China to we can go as far as Egypt and in Europe, Rome, and from the sea we can go almost to I think over Indian Ocean and to Africa. So this is the Silk Road along the land. In old China, and people they use animals from most of Chinese land and to different ways, maybe 3 or 4 ways. One is to I think nowadays Uzbekistan, Kazakhstan, and another middle way is the Far East. We can go to Turkmenistan, to Iran, Iraq, and Turkey, and then Istanbul, and some other ways we go to Italy. So it is very far. And from somewhere from Iraq, we can also go to Israel, Jordan, to Egypt. So this is a very long way. I think the Silk Road is international, regional goods exchange for many different kind of different people from different places. And this is a picture, and we still can see people use camels to carry the goods to exchange. In old times, we do this as well. Okay, the Silk Road, and the first name was named by a German traveler in 1877, and in 1938, a Swedish geographer, he published a book called The Silk Road, and this is very old. And as we can see, this is a picture from the book, and the goods carried by the camel is clearly can be seen. And this is the horse itself. As we can see, there's rope, I think, and the other end is the animals. So, in China, we have not only we have some of the old transcripts of the Silk Road, and it began as far as the West Han Dynasty, and it's about almost 2,000 years. And the Silk Road connects China with West Asia, Europe, and Africa. And because, as we know that China's goods is for the silk and our tea, and we got some goods from Persian, which I think is copied and other things. But very interesting is that most people in China we know are Persian businessmen, and they appear in China, some of the cities, because in old books we have some of the description of them. Not only in some common goods, we also have some of the commerce on traditional medicine, such as we talk about many times saffron. Saffron is very typical traditional medicine. Traditional medicine we got from Iran. And also in the book, Li Zhen, he also has very detailed description of this one, and where it was from, and how it was used, and who got this from the Persian. So this is very in detail in the book. And in previous years, we have invited some professors coming to the hometown of the author. This James is Qichun. It's about 3 hours driving from Wuhan. And if you remember, we have some Professor Alex is inside here. Only Alex. Okay, anyway, so I think it's a good experience. And we have visited his home as well. And this is in 2019. So let's talk about a few minutes with on the compendium of the material matter. And this one is compiled by Li Zhen, and he was born 500 years ago, and he devoted his entire life to combine this masterpiece. And maybe he used over 30 years since when he was 30. It's like to do this one. And this book is huge. It contains 16 sections. Different sections on water, fire, earth, metals, stones, and minerals, herbs, cereal, vegetables, fruits, food, fabrics and utensils, insects, worms and amphibians, animals with scales, shells, both animals and humans. So it is very interesting. Also includes human stuff for medicine. Right? this, it contains 1.9 million Chinese characters. So it is huge. And as we can see, we shown this just before. In English, is more. It contains six large books. This one connected 1892 kinds of medicines. And it has only 11,000 prescriptions. And it also made illustrations of over 1,000. In English, the English version was published in 2003, and it has over 4,000 pages in total. So these books systemically describes the knowledge of the traditional medicine, including the names and explanations and corrections of previous workers, and the quality of herbs and tastes, and indications, explications, prescriptions. I could everything he could know in his age. And he himself also visits many different places of China, and also he always comes out from different traditional practitioners, and to ensure its authenticity and accuracy. So this is a book by himself, but also he collects as much as possible in his age, but still it contains some of the errors in that age, in that times. And this book was also once praised by Darwin as ancient Chinese encyclopedia, and also has been translated to many different languages. In 2011, it was documentary heritage, and also selected as Memory of the World Register. So it is well known in Chinese, and I have our friends when we talk about Chinese medicine, we remember this book. And as I just mentioned, if it's possible, I want to send a copy of this book to you as gift. I will try my best to do that in the future. Okay, so as I just mentioned, we want to know there's something medicine from not only in China but also from other countries. So in the book, people has noticed that 96 medicines were from other countries, and among these 46 clearly mentioned from Persia. So that includes 9 herbs, 12 wood, 7 fruits, 15 vegetables, and 1 bowl. So I'm going to go through. Before this, I will try to just one. open up Chinese culture to the West as well. Okay, now I'm going to introduce quickly of all the medicines that we have in the CMM book. And the first one is natural indigo. And this one, it contains 3 different herbs. I put 2 figures over these 2. And it used to be a dye for the clothing, but now, even now, this plant was very commonly used for the medicine. And it treats the vexation and fever, and in general, we think it can inhibit the inflammation. And also, this one is formally in our pharmacopoeia book, and we culture this one a lot in large area. So this is a few examples. I think we have companies to make this kind of medicine. At least 50 different companies make the medicine like this. So this is very important. And also, as you know, that we have the COVID-19 endemic in COVID-19 around the world, and this one, this medicine is the very common one that people will use to treat the virus. Maybe we don't have the clinical result for this, but people they always like to take this one. It is a OTC medicine, so it's very popular and we use it in large quantities. And the second one is long pepper. Long pepper is not very common, and in the book he says the soup, boiling of this one with milk is excellent in treating the headache, rhinorrhea, and toothache. It grows like this. I don't recognize this plant. And this one, now China we have a small area of cultivation, but mostly we import from Indonesia, Philippines, and Vietnam, and we no longer import from Iran. Maybe my friend, if you can find a way to in Iran, maybe that's another resource. Anyway, we need 700,000 kilograms a year, and we use it in some of the products for pain reliever, tooth pain, joint pain, and digestive function. This is this one, the second one, and the third one, fig water flower, and this one is the dry resin. It is very close to, in Chinese name, very close to the plant just that the lady, Professor Ye was mentioned, but they have similar treatments with anti-inflammation and antibiotics, but this one is we use this very less than that one. Anyway, in TCM, we think this one can tonify fever and the gallbladder, groups ISS, and our treatments. So in this plant now is mainly used in Tibet and other tribes medicine. So it was no longer as the national white medicine nowadays. The fourth one is Venus, almond fruit, this is dry and mature fruit from each plant. So they have some of the chemicals, bornyl estates, camphor and bornyl. So this is a plant. Let's see. All these pictures were searched online. So this one, it is still included in our pharmacopoeia book with Chinese name, and it shows that the pharmacopoeia effects of the gastrointestinal protection and a lot of things like anti-inflammation, anti-diarrheal, bacteriostatic, and so on. But in south part of China, people like to use it as tonic food. That is when they have soup and porridge like this, they add this one to eat. Other places they may not do so. And betel. So this one was is a plant from the piper genus piper. So they believe that piper detoxifies toxins of miasma, and this first magnificent gas is resting with the chest, the warmth, the sleep. So this one, no, we don't use this one as medicine anymore. Number 6 is the Florella coryphellaria. English is hard for us, but the Chinese is the last 3 is Chinese characters. It is fructus, is dry ripening fruit of the plant. And this one, they have some wide range of pharmacopoeia effects, anti-tumor, antioxidant, anti-inflammatory, and something like this. It is included in our pharmacopoeia book. Generally have functions nourishing the kidney and invigorating treating skin disease such as fluorosis. And these two traditional formulas contains these plants. And jasmine is a beautiful flower, have very good smell. I think it's a very good landscaping plant. But it was believed to have something to clearing heat and relieving bacteria and draining them in our TCM theory. But it is people use it as tea, but it's not included in our pharmacopoeia book. Number 8, carpentium, broadenoi. This is from plant. It is still included in the pharmacopoeia book. The efficacy of clearing heat and resolving toxins. So this is the right we can see from the seeds, very small seeds. It is mainly used for the sore throat, tonsillitis, and other diseases. This one, number 9 saffron, is very famous one. So it is the stigma of the Crocus sativus. It is in pharmacopoeia book, but in general, people use this one as tonic food. Like in China, most people drink the tea as tea. But in the pharmacopoeia book, we said it can treat depression and tranquility the mind, activating blood and resolving stasis and cooling blood and other stuff. And I went to Iran, and the people make this one as this one to the rice as cooking spices. And eel. Eel is used for treating digestive system diseases. And this is a plant like grow like this. People now it's not included in the pharmacopoeia book. It's eel. And oh, sorry, I forgot to. We moved to the vegetable part. Vegetable now is vegetable. Cediado, this is a very common vegetable, and they have a rich carotene, vitamin, protein, calcium, irons, and other minerals, and very common vegetables. It was thought as it can do clearing heat, moisturizing dry lips, and invigorating blood circulation. Obviously, it is just a vegetable. And another one is the fruits, another kind of fruits. One of these one is onyx, date. I think I have seen this in Iran. Date is a fruit of the onyx, and it grows in high temperature area in deserts. This one, people think this can clear heat, more moisture in dry leaves, and as well. But it is not in our pharmacopoeia book. It is just fruit. And some of hot area in China, we also have this one as well. Another fruit is the fig. Fig is also a very common fruit, and it has very good nutrition. And yes, just the fruit. It's very common in China. We can buy in the shops, in the food stores. 14, Cassia fistula. This one in China is this plant. We just use this in the garden and the parks. I don't think I have seen this one to my attention. Anyway, it is mostly as a tribe medicine, like Tibetan binationalities in their tribes medical books. They use this one, not in general Chinese. So it is not included in our pharmacopoeia book. And pistachio, this is a very world famous fruit. And even now, I know that Iran make a lot of this one, and the other one make very good quality pistachio is USA. So I don't need to introduce this one too much. This is obviously just a fruit. And olive is also a very popular fruit, and it has a very good one. It's called olive oil, and it's called a Mediterranean diet for the cooking and good for the people's health. So this oil rich in antioxidants and other like polyphenols and health monounsaturated fatty acids. So this one is was the dry fruit was included in our pharmacopoeia book, but not now. This is olive. The next one is jackfruit. This is a very popular fruit in China. I think we mostly import from nearby countries, but like Thailand, Vietnam. So this one wood in old times. Right now, we can see everywhere in the street. And obviously, it cannot be as medicine. The grapes was also included in the CMM book. So it is, as we can see, it is just a common fruit nowadays. 19, Chinese eagle wood. This is a wood. This is now we moving to the section of the wood. This is the wood. This is from the. Sorry. This one is the wood of a very special kind of trees. One kind is called, we call it, this one we call it Tenxiang in Chinese eagle wood. And the people, this wood is very expensive. People use it to make different jewelries. And it has a very special smell. And the other one is this one, same one. So this is sorry. This is a very close one, and we make the smoke. This is the product. Normally people use this one, and this one, it is included in our pharmacopoeia book right now. The next one is called balado. It's a tree, like looks very close like a Chinese willow tree, and it warms, it warms the interior, purifies the waste and the kidney, dry hair and beard, make beard black. So this one, it is not included in our pharmacopoeia book. And the next one, myrrh. Myrrh is a very typical medicine from Persia. So it has from this resin from the two plants, Commiphora, and the other one is Commiphora momo. This one, we still use this one, and people think we put blood stasis, animates swelling, relieve pain, and promotes muscle growth. As we can see, this is the resin from the tree directly. So, in our pharmacopoeia book, we have natural myrrh and coordenador myrrh. They are from two different trees. So this is very interesting. I should look into this one in more detail. The other thing is this is very interesting. I don't know how much myrrh or how often we can see this one from trees in Iran, but right now China imports this one every year from Ethiopia. Ethiopia is more stable in politics, and sometimes we got some from Somalia, and we still need this one every year, and that's a very important medicine. We don't have China, not at all. We make a lot of pain relievers like joint pain, arthritis, like this too. So this is a very interesting one to look into. And the professor in Ethiopia, their group is working on this one because I think if China needs this plant, this vaccine, other countries like Korea and Japan, they need this one as well. So the next one wood is benzoin. This one is also a resin from a tree called Sterex tonicaensis. So this one is included in our pharmacopoeia book. And to be honest, I don't know what kind of medicine it can make, but it is in the pharmacopoeia book. The next one is ferula, Asafoetida. So this one is very interesting that I was proposing to do some collaboration work on this one. It was very famous as a typical medicine as antibiotics. Right now, this one, two things I have to mention that China, we reported two species in China in 1950s, but after maybe 20 or 30 years of harvestation, right now these two, we almost cannot find these two species in the wild. So right now all these ferula, I think we imported from Iran. Unfortunately, this ferula have very strong bad smell. People don't like this one. So people use antibiotics to replace ferula. This is the situation. But on the other thing is ferula, some of the people say that it can be very good bait for the fishing. Fishing is very interesting. Some people they want to buy this one still, so this could be some very good business. But I don't know how much we can do this, and if there's a way to do with ferula as a bait for fishing. It worth some money, at least. Aloe is very common plant. I don't know why it is included in the wood. Aloe is still in pharmacopoeia book. It's a dry product of the juice, concentrated juice from no species. So they have some effect of the sterilization and the bacterial, immunologically, and the tumor. I didn't see research on this, but I think we use more in the cosmetics. That's the right one. Some people even eat this plant as well. The other one is not goats. So this one is the system of a special larva parasiting on the young branches of the tree. Sorry. So use the unpierced means the larva still in their beds. I think used to use as medicine. So it was saying that it can strengthen the teeth and fixing gum, but it's not included in the pharmacopoeia book. The other one, frankincense. This is also resin from trees as well, Boswellia, two species. Also, this can strengthen the teeth. And also now it's commonly used in tribe medicine, still in our pharmacopoeia book as well. Oil is have very long history in China. So its crystals we obtained by steaming of the trunk, the trees, dry allen logs of the book. In China, it was always imported from other countries, but now we found trees after 1980s in China. So we have two kinds, borneo, and can also be, because chemically it is kind of simple. We have the ones from the trees, and also we can synthetic and made this one. So two were included in the pharmacopoeia book. And we have another plant, cinnamomum camphor, also makes borneo as well in China. It's also, this tree was found in 1980s as well. Two different trees that make very close chemicals. And Chablis, this is dry fruit. And this one is used for chronic diarrhea and dysentery, blood stools, and rectal prolapse, something like this. It is still included in pharmacopoeia book. Oh, the numbers. Another one is big fruit M, and this one products is made by fermentation and drying the fruit pot of the tree. I don't know. Never seen this one. It is not included in our pharmacopoeia book. And abele is a wood. It's a wood of some. It's very special wood used for detoxifying toxins and treating clogged with vomiting and diarrhea. Even though it is not included in our pharmacopoeia book, but the wood is very expensive. Some people they like the smell, and they would bid for very high price for this one because of it has to pass a very long time. Okay, now we talk about very quickly about metals, stones, and minerals. We have more in this kind. And metals, as we can see, silver, not in the pharmacopoeia book, but in every kind of list they have some treatments in old CMM book. They have some reasons to include as medicine, but obviously some are not correct. It effectively treating the disease. Some may have the toxicity problem. So silver not included in the book. And tacromite is a copper chloride. Is not included. And aluminum stone has many kinds, but right now we have 2 of the 5 still is in the pharmacopoeia book. Aluminum and malan tertium, 2 kinds still in this. And sulfur as well is not in our pharmacopoeia book. Salt sodium chloride, it is the food addictive. Sodium, and this one is the lead arch. This one is lead, mineral lead, so it's toxic. Iron in the pharmacopoeia, but not now. I don't know what kind of disease can treat iron and gold. Gold is not in the pharmacopoeia book. In the future, people use the gold foil, very thin foil, to treat the disease. And silver. People use silver as medicine in China for a very long time. For example, also as utensils like needles for acupuncture and mox busting. So silver, some of the metals are used for anti-infection, but silver itself is not included in the pharmacopoeia book. And the copper, metal copper. So copper is not copper iron, it's natural copper itself. It's recorded in the pharmacopoeia books. So for injuries and fractures and contusions and strains, because of the stasis swelling, we use this one. I think it's not very common. And the lead. So lead, people think this can do a lot of things. In old time, a lot of people they use some of the lead stuff for the improve their life. So it has a lot of treatment, but that in general is very toxic, should not be used. Coral is very coral from the sea. So they think the coral is similar to the gold. It's not included in the PTRC. And the other one, cinnamon. This one is mercuric sulfide mineral, and it's very good color. And a lot of Chinese old alchemy, they use this one. They want to live forever, and it can have some of the treatments. Cinnamon is still used in pharmacopoeia book, but not in general. We use this one very often. The other one, calamine. This one is the carbonate mineral, calcite, that contains about zinc carbonate. And this one is included in the pharmacopoeia book. It has efficacy to detoxify, moving eyesight and removing nebula and other stuff. And the other one is fibroferrite. And this one, it was long time ago. Song Dynasty used this one as medicine, but after that, it was not. And animals, ostrich. And this bird was from Africa, so we got this one 2,000 years ago in China, and I think Persian merchandise bring that into China. And obviously it is not in the pharmacopoeia book. So these tables, we shows the names of all these 46 different medicines, and we have the record of where it was from the CMM book. Some mention like the first one is Persia, and some it also have other places. And as we can see now, it's changed. And some of the current description after 2,000 years of exploration, and we may find other resources in China, in nearby country, and we don't have to travel to as far as Iran to get all these medicines. Some we may have where people may not use this anymore. So it is very interesting to do this one. Like saffron, we mentioned 2 places in this book, and now I think some places still grow this one, like Iran, Europe, and Afghanistan. I think people culture this one, and we have this one and 30s. And as we just mentioned, like 21 where we mentioned in the CM book, the old book, mentioned 2 places, Persia, South China Sea countries. But right now it's Somalia, Ethiopia, India, and other places. So we try as much as we can to make it more correct. But it needs multiple work to do this one. And this is the rest of this one. And all species now is in China. It's animals only shown in Zoom. So this is a very interesting. Some I think it's technology-wise. Even we say it is only from the book, like silver. We say it was only from Persia, but now in China we have this stone as well. So very interesting to organize all this. And the next one is the book. I just mentioned that the author is Zhen. He was born 500 years ago, and so the book is about 400, maybe 50 years old. But the 46 different medicines based from this book and other books, they were from they came to China at different times. Like the first one, different times before 220 years is about 100 years ago. We have 1, 2, 3, 4, 5, about 10 kinds, and 1,500 years ago, around that time, for non-10, and mostly 1,000 years ago, we have more. So this is very interesting. And after that, I think we have just very little, like silver and other stuff. So this is the thing we organized, and it is very interesting. And people can travel over 10,000 miles from Iran to China. And I went to Iran once. It is very, very long time. From Ulaanbaatar to Tehran, it took 4 hours of a flight. And travel may took 1 or 2 months to that. Okay, so I thank students, master degree students, Xin Zhou, she organized all this talk, and also I thank the Ministry of Science and Technology for continuous support for our programs, and thank you, and hope we can do more in the future. Thank you.
Thank you, Dr. Hu, for presentation. And I would like to thank everyone, the speaker this session. And now we have a tea break, and I would like to everyone to back here to next session. Okay?
Okay. Yes, I would like to mention something.
Okay.
Hey, do you have a question? Do you have a question?
Yeah.
Yeah.
Thank you, Professor Hu.
Okay.
Yeah, thank you, Professor Hu.
I'm sorry. Sorry. Please.
Professor Hu, thank you very much for reviewing all the exchanges between China and Iran. And most of them was true, but a few of them maybe a misunderstanding. For example, jackfruit, I don't think so. I don't think that Iran has kind of jackfruit. Maybe, I don't know, in the south of Iran. But this is the opportunity of having some kind of all the exchanges scientifically and economically between Iran and China and Iran and all the world and all over the world, because all the opportunities from our land, yeah. And I will mention some of your for example, indigo in my presentation as well. This is one thing that we are in touch from 2016, and I am now from the other professor collaborating with scientifically with other professors in China, but we couldn't do it with ourselves till now. I hope this goes in a good way in the near future. Thank you very much.
Thank you. Yeah. Hi, Jules.
Okay.
Go. I know the time is very limited. I spent a lot of time in this one. Can we, because my mistake, and I don't put any time break here. Can we take 10 minutes break, and then we start the next session? Okay.
Now we have a tea break. I would like to thank us for speaking this session. Thank you very much.
Thank you.
Was beautiful. Thank you.
Hi, Professor Annie. You may start the next session. Hello?
Yes, we have to start, Hugo.
Hi, Professor Annie. Now you replace Professor Sheng Wei as the host of the next session. Hello? Hi, Annie from Sophia University. Can you hear me?
Hugo, I hear you. I'm on a leave. But who is host have to open meeting. It's not visible now.
I was saying you to be the host. Hello?
Okay.
Sorry, Brad.
Okay, no, it's okay. Some problem was with connection. Okay, I hope we're here. Who is it okay? I will be host for this session. We will change it. Hugo, can we start?
Please. Yes, please.
Okay. I hope you'll take rest after our first session. We have very interesting lectures, and it was good last lecture of Professor Hu about Compendium of Medica and China Pharmacopoeia, and now our first lecture is going to Professor Alexander Shikov. Welcome, Professor Shikov, who is from St. Petersburg State Chemical Pharmaceutical University, and the topic of his talk is Medicinal Plants of Russian Pharmacopoeia, Regulatory Aspects and Use. It is your order, Professor. Professor Shikov, you can share your screen.
Yes, thank you. Just a moment, I will see how it works. With this, well, I need to
Yes.
screen.
Okay,
So,
always okay. Yes, you can start.
good morning, good afternoon, or good evening. Depends from the time zone you are. First of all, I would like to thank Professor Hugo for invitation to participate in this interesting meeting. This long time we were not communicated offline, face to face, and I'm sure that all of us just dreaming when this COVID restriction will be released and we meet somewhere, China, in Russia, in Bulgaria, in Iran, or somewhere else. Well, my today's topic is about the medicinal plants of Russian pharmacopoeia. Just one lecture before Professor Hu have introduced medicinal plants from Chinese medicine, and now is my time to present for you and to share with you some knowledge about medicinal plants which we are using in our Russian medicine, in our Russian pharmacopoeia. So, if you look on the map, of course you agree that our country is very large and we have a different climatic condition. Here in North Caucasus we have subtropical, here in North altitude Arctic. So you can expect that we have very broad biodiversity of medicinal plants, probably more than 21,000 of plants are native for the flora of Russian Federation. Unfortunately, among them only about 3,000 plants have been studied and some active compounds have been described and identified in this. Actually, our population is very specific regarding medicinal plants, and even it can be regarded as herbophilous society because more than half of our population uses medicinal plants on regular basis for time to time, treat different diseases or for prophylaxis of different diseases. The history of traditional medicine and use of medicinal plants starts with special knowledgeable experts, which we call in Russian znakhat. I believe that my colleagues from Bulgaria could even understand this word natively. It exists, my point of view, in Bulgarian language as well. And all the knowledges were recorded in chronicles, special manuscripts. Actually, the predecessors of the pharmacopoeia in Russia was the book with the name Whole Vineyards. In Russia, we call this Прохладный Petrograd. And this book was written in 1672. More than half of this book was dedicated to the medicinal plants, and 340 plants were described in this book. And the history of our official medicine and our pharmacopoeia starts with the Katerina the Great, the doctor of very famous our Caesar Peter the Great. Katerina have established an imperial medicinal collegium 1763, and 2 years after this, the first official military pharmacopoeia Astrensis Rassica was published. Of course, this first pharmacopoeia was not civilian, it was dedicated for military reasons. At the time, Russian Empire was involved in different wars around, and it was very important to synchronize and to unify the approach for treatment of soldiers in different regions. So, the first civilian pharmacopoeia
1978. And this pharmacopoeia was written as it was accepted during that time in Latin language, but this was the first civilian pharmacopoeia Russica. And among 770 monographs described in this book, 316 were dedicated to herbal medicinal preparations. On line 29, sorry. My slides was lost. I just like to mention that our pharmacopoeia is one of the oldest official pharmacopoeia around the world. For example, USP pharmacopoeia was established only in 1820, more than 50 years, about 50 years later after our, and the pharmacopoeia, British pharmacopoeia, was established in 1864. So my country is among the countries which became the official pharmacopoeia in the first life. At the moment, what is the current situation? In May 29, 2015, the Russian Federation, Belarus, and Kazakhstan have signed the Treaty of the European Economic Union, and later Armenia and Kyrgyzstan joined this union, and altogether we have the market for about 180 million of people. And for this reason, we have developed, and even we still work under unified, carbonized pharmacopoeia of the Eurasian Economic Union. That first chapter of this pharmacopoeia was published in 2020, and now we are waiting others. If you're interested to find some information, really online information and actual information about medicinal plants and other medicines used in Russian Federation, you can use this online service. Let's see, this is the link which you can access, and only one probably limitation of this database is only in Russian language, so you have to use it to Google Translate or something other. And then you can write the name of medicinal plant or name of formulation, and you will get all current information about medicinal plants or any other medicines used in the Russian Federation. Altogether, we have on our market about 25,000 of medicinal preparation, and it's not bad to my point of view. It is really not bad that about 18% of this list is dedicated to the herbal medicinal preparation. I must to mention that herbal medicine is a part of official medicine in Russian Federation, and therefore have the same regulatory approach for herbal medicine as well as for synthetically derived or microbiological or vaccines are and population and popularization during the last years. So even radiopharmaceutical preparation, all are regulated on the same basis. So starting from 218, Russian Federation follows to a new 14th edition of State Pharmacopoeia. This book looks like this, and it comprises 4 chapters. All information about medicinal plants is summarized in 4th chapter, and for medicinal plants and herbal material we have
special general monographs. The first general monograph is medicinal plant raw material, herbal pharmaceutical substances, and we have special definition for herbal medicinal products. The first medicinal plant raw material is fresh or dried plants or part thereof used for the manufacturing of medicines by industrial organization or by pharmacies, veterinary pharmacies, or individual entrepreneurs. The next group is herbal pharmaceutical substances. These are medicinal plant raw material, as well as substances of plant origin and or combination thereof, products or primary and secondary metabolism of plants, including the obtained from the plant cell tissue culture. So even biotech products produced by herbal plant cell tissue culture, they are regulated under our pharmacopoeia, and we have in our market modern drugs. So additionally, these herbal pharmaceutical substances include crude, complex, or biological active substances from plants, products obtained by extraction, distillation, fermentation, or using other methods for processing raw plant materials. And the other group, which is described by these general monographs, is herbal medicinal preparation. Is medicinal product, medicinal produced or prepared from one species of raw plant material or several species of such raw materials. I would like to note that for medical species, we have a special separate monographs, each all medical species, and medical species are mixtures of two or more kinds of medicinal plant materials, which include integral parts, food or powderized parts of the plant, possibly with the addition of substances of mineral, synthetic, plant, and animal origin. Well, in this fourth chapter of Russian Pharmacopoeia, at the moment 107 individual monographs describe raw plant materials, among which we have one alga or seaweeds laminaria and one fungi, very famous and I believe known in China as well in Europe. This is the Inanotus obliquus or chaga. The list I show you the list of all medicinal plants which are described in our pharmacopoeia. But due to the lack of time, I cannot do point your attention in every one of these plants. Just prove, and then I tell you where you can find more detailed information. So this is a long list. And more detailed information you can find in the first hour article, which was published in 2014 about medicinal plants of Russian Pharmacopoeia and their history and applications, and the extended version was published in the beginning of this year and dedicated special to medicinal plants from the 14th edition of Russian Pharmacopoeia. So you can see the link to this article at the bottom of this slide. Then just 2 weeks ago we have published big review about the medical species, and in this case, focused only in medical species used for management of diabetes and related disorders. This is the journal Frontiers in Pharmacology section of the Ethnopharmacology. Again, at the bottom of this slide, you see the direct link where you can access this article and get the information. If you compare the medical species concept and the concept of traditional Chinese medicine, you will find a lot of similarities. And together with Professor Hu and the large team of colleagues, Professor Hu and from China and other countries, again in the same journal, Frontiers in Ethnopharmacology, the Frontiers in Pharmacology section of Ethnopharmacology, very interesting article which just describes the concept more detailed, the concept of traditional medicines on the example of Chinese Hu cleaning herbal drink. So if you're interested in this aspect, please look this article. We have a very interesting group of medicinal plants in Russian pharmacopoeia. Each word described in all editions of Russian pharmacopoeia. They just were referred in the first pharmacopoeia Rosica, and during all years 10 plants, they were mentioned in all these pharmacopoeias. These plants are Altae, Sinalis, Cassia, Putifolia, Frangula, Alnus, Juniperus, Chamomile, Menta, Pinus, Salvia, Ilia, and Valeriana. For European countries, the plants are very well familiar and very well known. I think that in traditional Chinese medicine, pretty all the plants use it as well, even perhaps the same plant parts use it in TCM or in Persian, other traditional medicine as well. If we see which pharmacological groups, which disease could be treated with the medicinal plants referred in Russian pharmacopoeia, we will see a lot of pharmacological groups, expectorants, diuretics, astringents, and so on, so on, so on, so on, so on. For some plants, we have very interesting position, like this position, when the activity is not clearly claimed, like this group 5 plants. It means that the plants have more or less universal usage and use it for the treatment of different diseases, and therefore pharmacopoeia don't limit it, don't claim it only one main disease for these plants, but they are more or less universal. And the largest group of these plants is expectorants, and very good example of we call this like a classical expectorant, these plants, which I think known very well for all of our participants. This is Licorice. It's used not only in Russia, but in traditional Chinese medicine. It's used in medicine system, medical system of other countries. As far as I know, in China, this plant have even the second name, like the Prime Minister of traditional Chinese medicine. And it's interesting, I would like to point your attention that recently, together with Professor Hu, published this very interesting article about the use of licorice according to this modern research possible to use not only underground part, the roots and rhizomes of Glycyrrhiza, but the lower ground part, on the aerial part of this plant, and the composition of biological active compounds very similar, and we believe that utilization of this aerial part will extend the use of this plant and will be the positive moment for full utilization of the health properties of this plant. And now I would like to point your attention in this group of plant, tonic and adaptogens still have this double name, tonic and adaptogens, but we prefer to call this group as adaptogens. And why I would like to point your attention in this group, I just show you two slides about the growing of interest group of these plants. Yes, unfortunately Google is not available in China, but in other parts of the world you can check this. This screenshot was done in October of 2019. It was the time when I have visited China and joined Expo and other international group, and we spent a very nice time in Wuhan. And you see that that time number of link in Google about adaptogen was about 2 million links. But just 1.5 years after, 2021, the number of links was almost double increase, up to 5 million plus links. So it's evidence about the increased interest of global auditorium and global specialized all around the world. In the end of 2020, together with very international group of specialized from Sweden, USA, Germany, Russia, India, UK, China, we have published this review, which is dedicated to evolution of the adaptogenic concept from traditional use to clinical systems, and review was focused in the pharmacology of stress and age-related disease. In this review, we have claimed the next term for adaptogens. Adaptogens are natural compounds or plant extracts that increase adaptability, resilience, and survival of organism to stress. Which pharmacological effects are attributed to adaptogens? Of course, this is a stress protective, increase of concentration, performance, and endurance during fatigue. Improves some aspects of mental health and social functioning. Antioxidants. This is resistance to the high sickness. Antioxidants, treatments of impotence, normalization of sleep, improving of appetite, improving of night vision, improving of quality of life. And adaptogens are important in age-related disorders. So for global population today, it's very important to extend active life. So adaptogens very useful for these reasons. Which plants belongs to the group of adaptogens. The most of plants were actively investigated in before aging of 20th centuries, and now there's a plants under active investigation. You can see a lot of publication in the literature about the plants, and groups of scientists from different countries working under investigation of adaptogens. So the first plant is Aralia elata. I know that it's very known in China, as well as most of other plants which I will show for you. The second plant is Panax pilatus. And of course, this is Leodococcus and Panax ginseng, which are known, I think, all over the world. People know what is ginseng, and a lot of people are known about the Leodococcus. The next plant is Levzea Rectamoides. Not my point of view is underestimated plant, and it requires more additional investigation because we don't know all the secrets of this plant, and it's very potent. This next one is Sedum roseum. This is a new claimed and accepted name for well-known Rhodiola rosea according to past classification system. This is the Sedum roseum. And Schisandra chinesis. Even the second name of this plant, chinesis, evidences about very high popularity of this plant in China, and I know a lot of DCM which include it. Well, active compounds has been identified, and I would like just point your attention not on the broad spectrum of this compound, but on the similarity of this compound with our internal metabolites, which plays, or even metabolites, it's regulatory molecules which play a very important role in our everyday life. So if you compare the typical structure of intensinoids, you will see it's very, very similar to cortisol. If you look on this pyringine, this compound from Eleutherococcus, it's very similar to levodopa. Look to this compound, this compound from other plants. It belongs to the group of epoxy fatty acids, aldehydes, and it's very, very similar to azolamines. So, if we discuss more deeply this question, we can understand that the regulatory molecules, they exist in the human body in very, very low concentration. And when we discussing about the high concentration of active compounds, especially plant-derived compounds, it's difficult to isolate the big amount of these compounds. I would like to ask you, do we need every time to increase the dose of active compounds? If these compounds work on the level of regulatory molecules, the concentration of such molecules cannot be very high. Our human body was created by the God that we need just a very, very few amount of regulatory molecules to have this regulatory function. Therefore, this is one of the explanation of high efficacy of adaptogens. And if we look to the mechanism of activity of adaptogens, they work in extracellular signaling and intracellular signaling. More rigorous and more biological pathways you can see in this review dedicated to adaptogens. for future investigations. So for the conclusion, I would like to say that medicinal plants used in the treatment of broad spectrum of disease. The efficacy and safety of these plants are endorsed by long-time application not only in traditional medicine, but even in official medicine. Just remember, for example, the 10 plants which I mentioned, which were introduced in the first hour pharmacopoeia official pharmacopoeia. The scientific category of adaptogens is considered as a novel term to describe the next generation of medicines. Why? Why this is the next generation of medicines? Existing pharmacological categories such as analgesic, antihepatic, antibiotics, sedative, diuretics, more or less assume a disease state which to be corrected through the drug action. However, the future of medicine, I believe the future of medicine will look the disease before it has changed, it has the chance to develop some disease. They will be no longer as anti-this or anti-that. Something protectants. There should be something protectants, like hepatoprotectants, cardioprotectants, neuroprotectants, and so on. They will not only protect the existing state of the body, but will strengthen as required by our duties at the moment. So the future information you can find in these publications, and thank you for your attention.
Thank you, Professor Shikov. Thank you for your very interesting lectures. Of course, your very nice and amazing articles, well known for us, for Russian Pharmacopoeia. And I wish to ask you something. I thought when I read your articles before, but do you have any idea, do you did some comparison between Russian Pharmacopoeia and European Pharmacopoeia? What is the percentage of overlapped plants?
Well, we work now under this harmonization procedure between Russian Pharmacopoeia and European, especially in aspect of this Euro-Asiatic, our Union and Euro-Asiatic Commission, which include different countries like Belarus and Kazakhstan, Pakistan, and so on, so on, so on. But nevertheless, we have some specificity in Russian, which is a little bit not completely same like the specificity of European Union Pharmacopoeia. One side, this is administrative question, other side is the question, perhaps this is the question of time. And one day it will be harmonized.
I hope in near future. Yes, also I know well your website about Pharmacopoeia, and I use it, but I think you have to, in your regulatory system in Russia, to some problems with terminology. For example, between Pharmacopoeia, Russian Pharmacopoeia, for example, biodiversity law, herbal substance, herbal preparations. Do you have like that problems?
Yes, the question of terminology is always exists, and the roots of this problem is that Soviet Union a long time was an isolated country, and we have developed our science independently from the European society, from the world society, which is based on English language. Therefore, we have a lot of terms which cannot be very simple translated to English. And otherwise, vice versa, there are plenty of English terms which cannot be directly translated to Russian.
Yes.
And this is one of the key questions of harmonization. You're right.
Yes, I'm asking because we did some changes in our terminology in Bulgaria after Bulgaria became member of European Union, but they have again same problems. It is very big problems. And one more question about how is Russian pharmacopoeia compared to changes in Angios Pamphylogeny group taxonomy system? You show Sedum rosea, for example, it is new name. How you change a name? It is harmonized, actualized, or you use the old system? For example, Yola rosea is old. Yes, white flowers, yes.
I see it.
Okay.
I see it.
Okay.
I would say that Russian is very, very conservative country. If you see, we have only 14 edition of our pharmacopoeia. Even we have very long history of our pharmacopoeia starting in 1765. So it's long procedure. And I presented this new name for Medicinal rosea like Sedum rosea because this is a scientific accepted name. And when we publish the papers in scientific journals, we have to follow the current rules of the plant names.
Yes.
As it's held by this database, the plant list.
Yes.
Before. But of course, in the pharmacopoeia monographs, we have very old this accepted name, Viola rosea. And make a change in the pharmacopoeia monograph is very, very long history. Perhaps only when the next edition will be issued, then we will be able to change the name.
Yes, thank you. Thank you very much. Dear colleagues, do you have any question to Professor Shikov?
Yeah, I have one question, Professor Aneli. May I ask one question to Professor Alex?
Okay.
Hi, Professor Alex. This is Farooq from Bangladesh. How are you?
Okay. You are okay.
Hello, Farooq.
To me, coming with Farooq, yes, it is your order. Okay, you can ask.
Alex, how are you?
Thank you. Fine.
Okay.
What about you?
Yes, I am fine. Thank you. It's a long time I have seen you here. Two simple questions. You mentioned here the total number of species that you have recorded in your country, 2,100, right? 2,100 species you have, plant species. So my questions, number one is that at least 2,100 species consisting of gymnospermic, angiospermic, rhizophytes, pteridophytes, algae, all, the lower plant groups to higher group plants. And number question two, how many medicinal plants you have recorded out of 2,100 plant species?
Well, I am very sorry. I have very low the voice volume. I don't just listen your questions. Could you repeat? I just increased the volume. I switched it to my big.
Okay, it's okay. Now you can hear me. Okay.
Oh, now it's better.
Okay. Sorry. So my question is that you have 2,100 plant species in Russia, right?
Yes, correct.
So 2,100 species, those are from lower groups, upper groups, gymnosperm, angiosperm, rhizophytes, pteridophytes,
the 21,000 plants, this is the variety of different plants. And we describe it very little. We have described only 3,000 of plants among those.
Another question is, another question is how many 21,000 plant species you have? So, how many medicinal plants? How many medicinal plants you have in your country?
Well, we consider medicinal plants only to the plants which included in our pharmacopoeia. In our pharmacopoeia, at the moment, we have 107 plants only. All others are just used in, they could be used in traditional medicine. The specificity of Russian, we have no separate herbal medicine. We have just one official medicine, and this official medicine means that plant, if it's included in official medicine, it could be sold in pharmacies, it could be used for the preparation of any medicinal formulations. But all other plants which are not in pharmacopoeia, they are not legible in Russian as medicinal plants, just traditional medicine.
Okay, I got it. Thank you, Alex.
Okay, Farooq, thank you. Give me a break a bit. Okay, do you have more questions? Somebody? I cannot see. Okay, Professor Shekov, thanks again. And we are going to next lecture is holder of Master of Pharmacy, Assistant Professor in Sofia University, Ivan Wawowski. He's my young colleague, just started his scientific work. Ivan, you can share your...
Yes, just a moment.
Okay, thank you.
Professor Shekov.
Yes, I must do switch somehow. How I can do this? Is it closed now, my window or not? Not?
I think you should turn off your video.
Yes, I try how to put this. I don't know how to do this. Select content to share. I have not any share.
Okay.
This program, I'm sorry, this program is a little bit new for me. I don't know how to change.
Okay, I think this is okay. Now Ivan can share his screen.
Yeah, give me a sec.
Sorry, thank you very much.
Do you see the presentations? The presentation on your screen?
Okay, now it's okay when you can see.
Yeah, okay, that's great. In the next 30 minutes or so, I will be bringing the subject of herbal medicinal products, ADC called R, respiratory system, applicability of traditional medicine in OTC drugs. First things first, let's say a few things about medicinal products. They can be separated into two groups based on the dispensing regime, whether or not they're prescription-based or OTC. The prescription-based, as you well know, require a prescription in order for the patient to receive the medicine. The other group are over-the-counter drugs, better known for their abbreviation, OTC drugs. I mentioned in the topic herbal medicinal products. Bulgaria, being a member state of the European Union, has to follow the legal framework for registering herbal medicinal products, and it has three regulatory pathways of registering herbal medicinal products. If a company wants to bring such product to the market, it has the option to register it as traditional use. To use the option traditional use registration, well-established use registration, or standalone or mixed herbal medicinal product. The first option, it has specific requirement. The product can be administered to the patient without medical supervision, aka it's OTC product. The patient doesn't have to go to the doctor, and it excludes administration via injection of this product. The other two groups, whether or not the future products are going to be prescription-based products or OTC products, that depends on the competent authorities, the Bulgarian Drug Agency or the European Medicines Agency, depending on the type of procedure. The products that are studied in this research fall into one of these three categories, as stated in their summary of product characteristics. The role of OTC drugs, they pose some advantages over other or over prescription-based drugs. First, they can be used to treat minor to moderate symptoms. If the patient has minor complaints, he can use OTC products to heal and not go to the doctor. They're easily accessible. They can be bought, as said before, without prescription from a pharmacy or a drugstore. And this way, you're bypassing the visit to the doctor's office, which in the case of a pandemic, as we well know, that's a viable option. You don't have to. The patient doesn't have to have a contact with potentially contagious other patients. Let's talk about the ATC. What is the ATC? The ATC is anatomical therapeutic chemical classification. That's a system that classifies active substances based on the system or organ they target, and their properties being therapeutical, pharmacological, and chemical. Why we chose ATC called our respiratory system? Well, it's simple. Respiratory complaints are quite often they can manifest throughout the whole year. With cases of flu or cold, like I said, the pandemic. And here on the slide you can see the different sublevels of this cold R. There are shown different preparations that fall into these sublevels. For example, nasal preparations, throat preparations, drugs for obstructive airway diseases, cough and cold preparations, antihistamines for systemic use, and other respiratory products. The aim of the study is to evaluate the way the traditional medicine plants are represented in the over-the-counter herbal medicinal products in the form of herbal substances and or herbal preparations. In order to do that, to reach this aim, a few tasks have to be completed. First, OTC products have to be characterized. What do we need to characterize about them? What percentage of them are actually herbal medicinal products of these OTC products? The different sublevels of the ATC code they fall into, and different pharmaceutical forms they are distributed into, and last but not least, how many plants, part of the Bulgarian flora, are used to produce these OTC herbal medicinal products. Having done that, the next step is to study some traditional medicine sources in Bulgaria and do basically the same thing with the preparations mentioned there. And the third step is to make a comparative analysis between the two bases of data that we gathered. Here are the sources that we've used for this study. On the left side, you can see the list of medicinal products without prescription in the Republic of Bulgaria. That's a list you can find on the website of the Bulgarian Drug Agency. The Bulgarian Drug Agency runs this list and periodically updates it, like 4 to 5 times a year, something like that. From this list, we've gathered information which products are registered in the ATC code R respiratory system, and which one of them are herbal medicinal products, and what do they contain, what type of extracts, herbal substances they contain. The traditional medicine books that we used, there are two books. Folk Family Doctor, as you can see, it's the 8th edition, 1934. That's way back, and that's 8th edition. So we can assume that the first editions are like in the beginning of the 20th century or the end of the 19th. So it's pretty old and gives a good view of like almost a century back what was the situation back then. And the other is relatively new. It's published in 2013, so it's a bit more new than the first one we mentioned. In this slide, you can see the percentage of OTC, the percentage of herbal medicinal products of OTC drugs with ATC code R. From this list, we've gathered information that are 227 OTC products that are registered with the ATC code R, and around 18% of them are herbal. The other 82% are not. These 18% are, I think it was 40 herbal medicinal products in total. On this slide, you can see the distribution across the different sublevels of the ATC code R. As you can see from the pie chart, there are three that stand out as a majority. The expectorants, the combinations, and the Hedera helicis folium extract. Those three form like 2/3 of the 40 herbal medicinal products, and they're all used to treat different types of cough, which supports my previous claim that the cough is a relevant disorder, a relevant symptom complaint throughout the whole year, as in case of cold, flu, and other diseases. The next slide, as I told you, we can see the distribution across the different pharmaceutical forms. Again, one major group we see the syrups, 35%, followed by tablets, pastilles, lozenges, capsules, and oral drops. If we are to interpret these results, this could be due to a few reasons. Syrups, all those pharmaceutical forms, they're usually used to distribute medicines that treat different types of cough. So it's not a big surprise that the syrups are so well represented, so to speak. The others, the tablets, the capsules, and the others I mentioned, they're also reasonably well represented, and that could depend on the patient, the group of the age group of the patients. For example, children, it's easier, more suitable to use syrups, while adults usually perform tablets, lozenges, or capsules. They can easily take while they're at work. So we can assume that that's the case here for this distribution. And mentioning the children group, I will speak about the pediatric group in the end of my presentation. As I said, how many plants are part of the Bulgarian flora? In those 40 products, 29 plant species are used in order to produce the OTC drugs. These 29 plants come in the form of herbal substances or herbal preparations. And 15 of them, which make up 52% of those plant species, are part of the Bulgarian flora, while the other 14 are not part of the Bulgarian flora. It's a reasonably big representation, we can say, but it still has potential to use more plants from the Bulgarian flora. Having gathered the information about the OTC products, about the plant species, the ATC code distribution, and the other indicators that I mentioned, we have to do the same with the traditional medicine sources. Here on this slide, you can see a table with information regarding the OTC products that I already mentioned and the traditional medicine preparations. As I told you, the plant species used in the OTC herbal medicinal products are 29, which fall into 26 genera. As I said, 15 of those species are part of the Bulgarian flora, and they come into these products as in the form of 21 herbal substances and 41 herbal preparations. The picture with the traditional medicine is a bit different. In the sources we found, there were records of 40 plant species spanned across 38 genera, and 26 of them, which is around 63%, slightly above, but let's say 63%, are part of the Bulgarian flora. We can interpret that as like a century ago, it was not just used medicinal plant species that are in the country, but there was a trade from other places. We haven't studied roughly where exactly from, but the other 37% are not part of the Bulgarian flora. These 40 plant species come in the form of 50 herbal substances and 33 herbal preparations in these traditional medicine preparations. The last indicator you see is a common overlapping species. Those are 13 species that overlap between those used in the OTC drugs and the traditional medicine drugs. In the next slide, I will show you a list of those 13. You can see maybe the Latin names ring some bells, or the English names. I've put in the English names, so you can, if you know one or the other better, you can remember some of them. Of those 13, only 3 are not part of the Bulgarian flora. The rest, the other 10, are part of the Bulgarian flora. There are some interesting examples among these plant species. I've stopped my view on 3 of those. The first one being Hedera helix, or ivy, its English name. In the modern day OTC drugs, it is used mainly as expectorant in cases of wet cough, mainly syrups, sometimes effervescent tablets or capsules and lozenges. But what we found in the traditional medicine sources is that there are no records of it being used as expectorant. It's not used to treat respiratory disorders. What it was used about is superficial wounds and pus wounds, to treat wounds, and nothing in common with the respiratory system. The other 2 examples that I want to point out are the primula and the ripwort plantain. So let's start with the primula. Primula sp species. It's found in Bulgaria, and in the OTC drugs, it is used Primula elatior, also known in English as oxlip. And in the traditional medicine preparations, it is used Primula veris, known also in English as cowslip. Both of those plant species are represented in the country. They can be found in the country, as you can see by those maps on the slide. They both have a monograph in the European Pharmacopoeia. As you can see, Primula veris It can be found across the whole country, and it spans from 0 to 2,500 meters altitude. While the Primula elatior, it's not so. It doesn't have that big area of distribution, and it's interesting about this species that it can be found at higher altitudes, up to from 2,200 to 2,800 meters. And another interesting thing about that is in the traditional medicine sources, this species, Primula elatior, it was not viewed as a medicinal plant. Maybe due to the fact that it grows at higher altitudes and it was not as available as Primula veris, for example. So we can say that that's the case. That's the case here. The other plant species that I wanted to talk about is the ripwort plantain. Again, it is used in syrups to treat cough. On the left here, you can see a picture of ripwort plantain, Pontago lanceolatum. You can see that the leaves are long and lanceolate, as suggests the name. They don't have big leaf surface. And in the OTC products, that's what's used, Pontago lanceolatum. In the traditional medicine sources, however, there is mentioned another species, Pontago major. That's broadleaf plantain. As suggested by the name, and as you can see by the pictures on the slide, it has much wider leaves and much bigger leaf surface. Again, it is not used to treat respiratory diseases. It was used to treat similarly to the IV, superficial wounds and hemorrhoids. And the more common of the two is Pontago major, because of this higher, larger leaf surface, and Pontago lanceolata was not mentioned as much as the other plantain. Here you can see we've made a research about how many components are included in the OTC drugs and in the traditional medicine preparations. We've set 3 different groups. Monocomponent, second group contains 2 to 5 plants, and the third group contains more than 5 plants. Let's see what we found about the OTC products. As you can see, 60% are monocomponent OTC products. 35% of them are they contain 2 to 5 plant species in the form of herbal preparations and herbal substances, and the last group is just 5% that contain more than 5 plant species. If we look at the traditional medicine sources, the picture is a bit different there. You can see 88% monocomponent preparations, 10% that contain 2 to 5 components, and just 2% that contain more than 5 plants. We can interpret that as a somewhat growing tendency to use more than one plant source in a OTC product or a preparation to attack the problem, let's say, from multiple sides. As you can see, the percentage of components 2 to 5 and more than 5 is grown, I would say, for the second group significantly from 10 to 35%. And as I mentioned earlier in the previous slides, we've considered the pediatric population. From the OTC products, 38 of them, or 95% of the OTC herbal medicinal preparations, they can be used, they can be administered to children in different ages, depending on the limitations of use and the contraindications of the products. What do I mean by contraindications? For example, you can see the structure of eucalyptal. The age HMPC monograph that's a list that is run by HMPC, Human Herbal Medicinal Preparations Committee. It is stated that if a product contains eucalyptol, it shouldn't be given to children that are younger than 30 months due to the risk of laryngospasm. If we look at the traditional medicine, you can see that the percentage is much lower. That's just 7% or 4 preparations that are used that can be administered to children. And those 4 preparations can be administered to adults also, but in higher dosages, as stated by the traditional medicine sources. But what we can make from this is that it's more oriented towards finding options to treat respiratory diseases in children. As in the past, like the past centuries, it was not that well presented, so to speak. And you can see that there are no overlapping species used for children in the traditional medicine and in the OTC products. So we didn't find any records on that. In conclusion, based on the found data and the analysis that we made, we can say that the OTC herbal medicinal products that are registered with the ATC code they are produced by a diversity of plant species in the form of herbal preparations or herbal substances, yet there is still some untapped potential in the traditional medicines that can be used to produce new products and bring new products to the market using the knowledge that contain these traditional medicine sources, being that with different combinations or just again monocomponent OTC drugs. And with that, I thank you for your attention, and I conclude my presentation. Thank you.
Thank you, Ivan. Thank you. Colleagues, now is your order for question. Do you have any question to Ivan?
I have one question. Can you share the slides with us, because I want to see. I didn't write down all the names of the herbs. It looks very
just give me a second.
Yeah, thank you.
You're welcome. I'm assuming that's the slide you're looking for.
Cool. Can you repeat your question? We cannot hear you.
That's good. I want to know these names, and it looks very interesting. So these plant species are in the EU, right? EU system.
Specifically in Bulgaria, some of them, 10 of them are part of the Bulgarian flora.
I see.
And some of them have monographs.
Maybe later you can tell us what's the how the EU pharmacopoeia is different from each countries inside of the EU.
Well,
yes.
You can finish.
They have a own one, or each country they have a own one.
The European pharmacopoeia is one for the entirety of member states. The member states can use their own pharmacopoeias as long as they're aligned with the European pharmacopoeia, and some of those have monographs in the European pharmacopoeia. Others are not yet. They don't have monographs yet. You can try searching for them the European Medicines Agency's website, more specifically the HMPC part. If you search the link of the plant species or the herbal substance, you can find if there is a monograph in progress being written or some assessment report.
I see. Oh, thank you.
You're welcome.
Thank you. Do you have more questions, colleagues? Okay, cannot see. Thanks, Ivan, for your interesting presentation.
Thank you.
Okay, and we can continue with our program. And now it's order about Dr. Heitor Ribeiro da Silva. I hope pronunciated generally okay. From Federal University of Amapá, Brazil. And the topic of speech is technological development of formulations containing standardized extract of euterpe oleracea.
Hello.
Hello.
Can you hear me? Can you see me?
Yes, we see you and we hear you. Yes, now we can share you.
Thanks very much. I will put my presentation. Just a minute.
Yes.
Okay, it's all right with my presentation to you?
Yes, but maybe to put in full screen. Because now we see a bit more slides. Okay, let's start like that, okay?
Is okay now?
No, now I cannot see your screen. Let's again.
Okay.
Okay. Let's start. Yes, now is okay.
Okay. Well, good morning for Brazil. I think in the other countries are good afternoon or maybe good night. I'm Heitor Ribeiro da Silva from Macapá, Brazil. I'm very grateful to the invitation to be participating in this very important event. Today, I'm going to talk about the work conducted in my doctoral thesis. The title of the work, the study was Technological Development of Formulations Containing the Ethanol Extract from Euterpe oleracea fruits. This work was supervised by Professor José Carlos Tavares Carvalho. Well, a little introduction. Much is now about the effects of the oxidative stress on the body when there is an imbalance of antioxidants and pro-oxidant species leading to oxidative damage. This damage results directly and indirectly in at least 15 known disease and disorders. Among them, we can see obesity, diabetes, neurodegenerative disorders, atherogenesis, and various types of cancer. Well, and so the idea to making pharmaceutical formulations containing acai extract was based on the fact that such plant is used and cultivated in the region, here in Brazil. It has high market potential, and a lot of information is available in the scientific literature. Biological activities, principally antioxidant activity, composition, and folk use. Despite such evident importance, it's hard to believe that in Brazil, no product takes good advantage of it. Therefore, in this study, we developed two different formulations of this product. The first formulation was a nanoparticulated polymeric system that could protect the product against degradation. For instance, anthocyanins, which are pretty unstable. In addition, this formulation could carry the active compounds due to its size and providing new pharmaceutical perspectives. The second formulation was tablets containing acai extract. This choice was because tablets have a high population adherence. In fact, 80% of solid formulations are tablets. They also provide good stability to the active compounds. So, the object of this work the main objective to develop formulations containing the ethanol extract of Euterpelera acimax. And to reach this main objective, it was necessary to obtain, characterize, an ethanol extract of Euterpelera fruits, standardize it for anthocyanins, in this case, obtain and characterize a nanodispersion containing the extract, obtain and characterize tablets containing Euterpelera extract, and evaluate the acute toxicity of this plant product. The material and methods were first of all to obtain the extract. A complete factorial design of 2 to the power of 3 with 2 central points was carried out to choose the best extraction conditions. The extract process was maceration, and a total of 10 experiments were performed. The independent variables were ethanol volume, acetic acid volume, and the time of extraction. The dependent variables were total solids, total phenols, and total anthocyanins. Just a minute. Okay. All those extracts were assessed for pH, density, and total solids, according to the Brasilia Pharmacopoeia. The determination of total phenols index was according to British Pharmacopoeia, and determination of total anthocyanins was evaluated by the differential pH method. Well, then the chosen extract was concentrated and lyophilized, then was performed moisture content and total ash assessment. From this point, the characterization was performed from just lyophilized extract. Well, to determination of mineral contents of this lyophilized extract was used an atomic absorption spectrometer from Shimatsu, Japan, equipped with an auto-sampler and a fast optimizer. The ashes was treated with 1 milliliter of nitric acid in a 50 milliliter flask. Was observed the amounts of calcium, iron, manganese, zinc, nickel, cadmium, aluminum, and lead. The HPLC chromatography profile was performed on HPLC UV Metsu class BP Japan, equipped with a high-pressure pump LC 2080 and auto-sampler CU20AC and UV detector SPD. The gradient was the gradient A, methanol, water, and acetic acid in these proportions. acetonitrile and acetic acid 9921 was started with 9525 AB and ended with 820 AB. The flow was 0.5 milliliters per minute. The total time of the run was 50 minutes, and the chromatogram was obtained at 520 nanometers. Also, were did morphological analysis by scanning electron microscopy, where the samples were lyophilized, examined under scanning microscope TM3030 plus Hitachi, and the determination of total anthocyanins content by the pH differential method and the DPPH radical scattering. The data analysis were performed by using the software design expert, also using analysis of variance with significant p-value and the adjusted value of R squared greater than 0.85. A convenience function of design expert software was used in this analysis, that analysis. To develop the polymeric nanoparticle system containing the extract, let's call it EEEO, was obtained two phases, organic phase and aqueous phase. The organic phase was prepared with 25 milliliters of acid solution, 15 milligrams of the extract lyophilized, 500 of elderdite with polymer. Under agitation, and the aqueous phase was obtained by 500 of 28 in 75 grams of distilled water under agitation. The organic phase were poured over the wet aqueous phase at a flow approximately 1 milliliter per minute under agitation for 20 minutes. To characterization this formulation was used as synthesizer 3000 from Malvern instruments and was determined the average size, zeta potential, and polydispersion index. Also did the conductivity and pH, stability by centrifugation, and encapsulation efficiency. To choose the best tablet formulation, the second formulation, to choose the best tablet formulation containing acai extract, 4 alternatives were tested with different amounts of magnesium stearate. The components were 20 grams of the basic granulates, patent protected, 5 grams of lactose, magnesium stearate, and 2.5 grams of freeze-dry extract. The process of choosing the best formulation was based on the physical-mechanical properties of each one. So, after choose the best formulation, it was compressed in a hydraulic press to obtain the tablets. The tablets obtained were evaluated according to the recommendations of Brazilian pharmacopoeia. The dissolution profile was developed under the following conditions. The dissolution was established with the quantification science using the pH differential method, released in the gastric solution by 60 minutes. The samples were collected with the sample of 5 milliliters were collected, filtered with paper. The estimate of the EEO, odor extract acute toxicity, was performed based on the OECD 423 class acute toxicity and guide for conducting preclinical toxicity studies of herbal medicines. Adult female Wista rats by 8 to 12 weeks was used. 2 groups observed, control group and treated group that received 2,000 milligrams per kilograms of the extract. The animals were observed by 15 days daily and every 3 days for statistical evaluation. Results and execution of the work for the extractive process. Observing the experiments involved in extraction planning, it was observed that the acetic acid volume was crucial for anthocyanins extraction. This can be explained by the flavulin cation in the extract that facilitates the extraction in acidic environments. Of the 10 experiments performed, number 3 was chosen to continue the work, although number 4 had higher values of the anthocyanins concentration. But the extraction time is needed to be consideration for the industrial scale. Also, the content divergence in both experiments is not significantly relevant. The characterization of the select dry extracts. The select extracts had the following values of pH: 4.1, moisture content, and total ash. All results are within the expected for a lyophilized extract. From a technological point of view, it's essential to know the content of residual water and volatile substance in the raw material. So that's the value that this value is considered in the yield calculations. About the mineral content, the determination of total ash allows quantifying the inorganic non-volatile residue present in the product and is often composed of carbonates, phosphates, chlorides, and several types of oxides. The elemental composition of the extract was also as expected from assay fruits. The sum of total mineral contents of the extracts is less than 20 micrograms per gram, which is the upper limit for heavy metals. In the HPHC, we can observe 2 peaks of anthocyanins. So this majority peak is anthocyanin 3O glycoside. So most of others researchers have similar results that we found it. Macroscopically, it could be observed that the extract had headstones, characteristic of the material rich in anthocyanins. In the scanning electron microscope, the extract had a porous and amorphous aspect. The lyophilized extract had 2.81% of total anthocyanins content higher than what is commonly reported in the literature. The inhibition concentration of the pH was 31.5 ppm. Finally, the formulations, the nanoparticle polymeric system, we can observe these results. The system was stable after evaluation of stability of centrifugation by centrifugation, evidenced by particle size, zeta potential, polydispersity index, and encapsulation efficiency. There's my mouth. Okay. Encapsulation efficiency. 48 hours and 30 days after preparation. So let's observe something about that. The pH value is slightly acidic, consistent with the composition of the active anthocyanins. By evaluation the pH in the function of time, some information about the nanoparticles stability can be obtained. For example, a change in pH can indicate polymer degradation. Zeta potential refers to the particle's surface potential. It can influence in the stability of the system and the incorporation of active compounds. In this case, higher the modular value, better the results. Generally, even we prepare it through different methods, nanoparticles have average diameters between 100 and 300 nanometers. However, we can see particles with average diameter of 60 and 70 nanometers, or even smaller than 50 nanometers can be obtained. So the qualitative composition of the preparation method of the formulations are decisive in the average diameter and polydispersion of these particles. Determining the amounts of the drug within the particles is especially complex due to their small size, making it difficult to separate the free drug and fraction from the particulate one. A widely used technique is determining the free drug concentration in the suspension after centrifugation. Next, the total drug concentration is determined through the complete dissolution on the nanoparticle in adequate solvent. Finally, the amount of the nanoparticulated drug is calculated by subtracting these two values. Notably, nanoparticulated polymeric system, this nanoparticulated polymeric system can maintain their characteristics even after 3 years of preparation. Finally, the tablets containing the extract. The chosen formulation was number 3 for presenting the best physical mechanical properties. The direct compression method was used because it's suitable for thermobio compounds and such as anthocyanins. Haeckel's graph proposed that porosity reduction happens in this graph. In this case, proposes that porosity reduction happens with the application of force. In the manufacture of tablets, it's convenient to have plastic deformation, forming tablets with good mechanical resistance. When materials are hard and brittle, they fragment, and when compressed, the generated fragments help it to fill the exit pores, aiding to consolidate the formulation. This slide shows the relationship between the friability and the hardness of the tablets and the compression force. This test assesses the mechanical resistance on the tablets to breakage. The friability test, the resistance of the tablets to abrasion in pressure is observed. According to the Brazilian Pharmacopoeia, the tablets must not show any cracks or breaks at the end of the friability test. The loss must be less than 1.5%. As the compression force increased, the tablets became harder and less friable. This integration test was also evaluated following the recommendation of the Brazilian Pharmacopoeia. In this test, the uncoated tablet should not exceed the limit of 30 minutes to disintegrate. Here, in this show, it is observed that all parameters are within the limit established by the Brazilian Pharmacopoeia. The tablets had an average weight of 250 milligrams or more, not exceeding 5% variation. Tablets produced with a compression force of 12 kilonewtons were selected as the final formulation. Despite having a high hardness value, the disintegration on an adequate time had friability values and mean weight with standard deviation and coefficient of variation according to the Brazilian Pharmacopoeia. Here is observed the dissolution profile of the tablets containing lyophilized acai extract. The result shows a high release rate right at the beginning. 5 minutes after the beginning, about 75% of the tablets and anthocyanins had been released. The values observed are following the authorized pharmacopoeias in Brazil. All those pharmacopoeias. As for the acute toxicity test, no toxicity was observed in the product. Then the extract, in this case. For the conclusions to the work, it was observed that the acid volume was crucial for anthocyanins extraction. Polymeric nanoparticle system had a high stability and good physical properties according to the literature. And the tablets had a fast dissolution and anthocyanins release. I want to thank you very much for your attention. I want to thank the organization for the event once again, and salute here the team of Pharmaceutical Research Laboratory, which is crucial for the development of our research. Again, thank you very much, and Carmi.
Thank you very much, Heitor. And dear colleagues, you have possible to make question to this nice presentation. Thank you for this interesting presentation. Do you have any question?
Can I ask one question?
Okay, Mati. Yes, okay.
Yes. Actually, this is it should be in my presentation as well, because I have also calculated the total anthocyanin in some sulfur petals. But just one question: in most of the cases, the total anthocyanin will be calculated as milligram per liter. Is it true? I mean, so this is because of the method, but if we would like to, calculate the milligram per gram of dry weight, what we should do? What do you think?
to facilitate the conversion to because we have to work with two formulations, right? The liquid and the solid formulation to facilitate the, how can I say, the explain. I did that information, but you're right. We calculate by milligrams per liters or grams.
Actually, this is my problem as well, and you know.
Sorry, I didn't understand.
My problem, I mean, I when I am extracting the anthocyanin, I would like to know that is it an alternative method for calculating the total anthocyanin or not?
My audio is too bad. Your problems having problems to calculate about the extractions.
Oh.
Sorry. Okay, we can talk about it later. Thank you.
Okay.
Okay, thank you, Amani. For another question to this lecture. I cannot see. Okay, Heitor, thank you. Thank you again for your lecture. And now we are going according to our program to lecture of Professor Hu to introduce to us Huazhong Agricultural University and the city of Wuhan, which are our host today. Yes, nice.
Hi, can you see me? Me again?
It's always okay, Hugo. We'll see. Maybe to put full screen. Okay. Thank you.
Okay. So now I think it's 5:37 in China. It's almost a time for students to come for dinner. Anyway, I will try to finish this one in 30 minutes. So I'm going to introduce our university, the city, and our work. So organized in 4 parts, I just mentioned. And the first one, following this 30 slide, was from the Dean of our University International Affairs. I got the slide from her. And I think she has she knows better than me of the university. So our university is an agriculture specialized university, and it is administered by the Ministry of Education. And we are among one of the 211 project university funded by the country. And we every 5 years we have evaluation by the ministry, and the 5 disciplines are the best in China in biology, including the crops, veterinary medicine, horticulture, and agricultural and forestry, economic and management, and animal sciences. And these 5 disciplines are major. They are not only good in fundamental research, and they also do very good translational research as well. So this is our motto: learn, practice, achieve, and help. And let's do something else. Okay. The record history of the university. Our university founded in 1898. That's over 100 years. And you may remember that in 1894, Japan raided China, and we have first China Sino-Japanese War, and China failed, and we lose a lot of treasure to Japan, and then we start to realize that we have to establish a good industry and agriculture to make the country strong. So a few years later, one of the commander in the area founded this farming school. It's the first one, and in the new country, 1952, we have a college, agricultural college. And 1985, we changed into university. 2000, and the university was transferred to the Ministry of Education. And Huazhong means central China. So it was one of the, I think, 8 agriculture universities belongs to Ministry of Agriculture. But only 3 was transferred to the Ministry of Education.